Don't worry.
They work on the surface, but on the surface you look calm and ready.
To drop bombs.
But you keep on forgetting.
But you're not. Yeah. I showered there. I had soap together.
But you wrote down. This is just talking to like us. We're just gonna go quick.
Aloha kakahiaka kako. Welcome to day 2 of the PIPES 2026. God, 2 days in a row. Our 2026 Intern Symposium. Mahalo for being here. Before we jump into our intern presentations, we just wanted to jump into a little bit about PIPES for those who are not familiar. And we'll start off with a little bit of staff introductions. Aloha mai kako, o ihono o Kiana Davis. He kiki no hopapa no kapa'au, kahi no nana pu'u hailelua o pili me kalahi kiola. Aloha, I'm Kiana from Kapa'au Kohala, and I'm currently a PIPES coordinator.
Aloha mai kako, o Leina Luinesa ko inoa no kaipapa'u o Ahumayau, and I'm a student coordinator.
Aloha, my name is Hinano no Puna Hawaiʻi Mayau. I've been part of the PIPES program since 2023, and I am a program assistant.
Aloha nui, my name is Viola Needham. I'm the PIPES program lead, and we're very happy to have our day 2 of our student symposium. I'd like to start off this morning with some mahalos. To these people that you see, but also to lots more, Lara Gomez and Ray Matsumoto, the dean of this pharmacy school. Mahalo nui for having us in your space for 2 days and allowing us to be guests here. I'd also like to bring in our writing team, our PIPES writing team. Forgot to mention yesterday, they've been an integral part of our curriculum and reviewing intern writing assignments and being a kāko'o to the program in that way. Mahalo nui to Corin and Aveao, Mia, Auntie Carla, and Corin at the Kilohana Learning Center. Our staff as well, that's been reviewing writing assignments. Mahalo nui for the extra hours that takes. To all of the ohana and other halves and the extended village that it takes to run this program for each of us here on staff. To our advisory board, Sharon Ziegler-Chong, Ulu Ching, Noi Punivai, and Becky Ostertag, thank you guys for having our backs. And to all the places and people and community that have hosted us and our hui, it's a small army that we have this summer. During the summer, it was wonderful to spend time with you and deepen our pilina. For our funders from Hau'oli Maoloa Foundation, Keahi Maikai Moku, Faith Paikukui, and Chelsea Chow, Marcelo Hansa and Sienna Mizuno from the University of Hawaii Foundation, Hualani Lu and Ho'olena Ioane of Kipuka Native Hawaiian Student Center here on campus, Summer Roper, and all the staff at the National Park Service, with which we are so fortunate to collaborate. Mahalo nui. Po'ala Pasqua and Maya Gudini from the Ahupua'a Accelerator, and the University of Hawaii Sea Grant under the Hawaii Conservation Alliance Foundation. Mahalo nui for your friendship and partnership. Janine DeFries from the Hawaii Pacific Foundation and the fabulous team that we were so fortunate to meet over the past 2 days. And the National Science Foundation. Mahalo for the tremendous support in providing research experience for undergraduate opportunities. Mahalo.
So as you all heard right before we jumped up on stage, we opened with our PIPES Oli Komo. This was haku'd in 2024 by our staff. And something about PIPES is we want to equip interns, those that come through the program, not just with technical skills, but really all the different things that are needed in their tool belt. And we think that Oli and protocol is just one of them. So you guys got to hear Oli this morning. And if you stay until the end of the day, you'll get to hear our mele mahalo. And one of my favorite lines is at the very end, Nu'a mai nā lehua I le kahiko ula ouka, vilia kalau ia I moku vai lehua. Which really highlights the intern and mentor relationship. We view you folks, the mentors, as the kumulehua. You're super pa'a in all of the backgrounds that you have, in the kuana ike that you hold, and the interns are really the ie that get to grow from and on the kumulehua. And hopefully by the end of today, you guys will actually get to see that bright red, orangey, yellow, beautiful hua on the ie, which is the product of all of the last 10 weeks that the interns have been working on, and the hua of all of just the pilina that has built over the summer.
Okay, I will be giving a brief history of PIPES that has been around for 32 years now. So in 1994, MASIP was established to create internship opportunities for undergraduate students from Micronesia and American Samoa, encouraging them to pursue careers in environmental science while gaining hands-on experience back home in their own island communities. Building on the success of MASIP, UH-HIP was created in 1997 to increase Native Hawaiian and Kama'aina representation in Hawaii's conservation workforce by connecting students with place-based internships through agencies and community organizations across the Pai'aina. With the support from NSF, Hilo launched the REU program in 2002 to provide more undergraduate students with research-focused internships that combine scientific research, community, and community. Over the past 32 years, PIPES has grown into a network of partnerships that has supported more than 1,000 paid internships, connecting students with experiences while helping build the next generation of Aloha Aina across Hawaii and the Pacific and the world.
So about 4 years ago, I believe, and from before that, this Mo'aina framework was birthed here at PIPES. And we call it an Aloha system of practice based in these 4 values and relationships that we believe are imperative to stewardship of place and people, at least here in Hawaii. Na'au, our connection to ourselves and the potential for transformation. Aina, our connection to our places and spaces, that which feed us and that which we can feed back into as well. Kaiaulu is our connection to community and the reason why we do the work and the impact that we would like to have on community. And ka'ao is our connection to our articulation, and there's an emphasis on storytelling here. So not just being a scientist, not just being a researcher, but becoming a really good storyteller so that the lessons from your experiences can carry on to the next generation.
The different pathways that we have, or the different pillars that Vai had just mentioned, are the different pathways that we have throughout our internship. The Na'au pathway really ties into offering students opportunities to go back to their places and remember generational practices, or just deepen that pilina that they have to certain spaces. Our Aina pathway is people who we like to say are the boots on the ground, fins in the water, who are getting dirty and doing the work on the day-to-day. Our Kaiaulu community pathway, much like it says, it focuses on community engagement and giving back to community and education. And then lastly, our Ka'ao research pathway, which, like we say, is very research heavy. So a lot of these internships look like they might be in the lab or just are very intense.
And then this summer, we had 35 interns placed with 30 different host sites. And this map just shows the spread of them across Hawaii Mokupuni. Each of the blue dots represents a space and a place that an intern got to care for and was hopefully cared by as well. And we encourage you later on to go and click on just each of the interns' pictures. Amanda will be starting us off this morning. But it takes you on a virtual huaka'i to where each and every single one of them got to be. And then our symposium schedule. Today is day 2, and Amanda will be kicking us off. Our lineup is pretty solid for today, so please stay tuned in person and online. Mahalo.
Hola, buenos dias. Aloha mai kako, o wau o Amanda. I'm from San Diego. How are we all feeling this fine morning? Okay, one more time. How are we feeling today? Thank you all for being here. I'm really excited to see everyone else's presentations as the day goes on. And I'm excited to just be here today for those that can't join us today. So thank you all again. Today, I am going to be talking about bridging ecology and community. I'm going to be talking about my research this summer of monitoring the Hawaiian wolf spiders and the factors that influence their presence up on Mauna Kea. So, who am I? My name is Amanda Campos. I am from San Diego. I was born in Pátzcuaro, Michoacán, Mexico, and was raised in San Diego and in the Bay Area. I am a first-generation student at University of California, Merced, where I'm studying ecology and evolutionary biology. I'm going into my last year, up and coming, so it's really exciting. I feel like all throughout my childhood, I've always been immersed in Aina, experiencing, exploring her, and just always getting my hands dirty, boots in the ground, somewhere being outside. I got the privilege of having a family that is very connected to their outdoor spaces. So it's been a privilege to just be outside and explore. And in college, I started getting more into conservation work. I work at Yosemite National Park back home. Shout out to those watching. And I've gotten the experience to not only work in conservation, but just see how that career goes and getting more of that, hands work. But I feel like all throughout college, studying ecology has led me to where I am today, applying for PIPES, which is a program that not only focuses on that ecology work, but also on that inner work that you have to do. This summer, I got the privilege of working with Center for Mauna Kea Stewardship. They're up here at the front. We're a pretty small team for a lot of the things that they do. And their mission is to protect and conserve the natural and cultural resources on Mauna Kea. So we work up at the summit of Mauna Kea. Some of the management areas that CMS focuses on is in that Halepohaku mid-level facility, the Summit Access Road Corridor, the Mauna Kea Science Reserve, and in the Astronomy Precinct where all the observatories are. Some of the ecosystem types up on Mauna Kea start at that lower elevation in the subalpine woodland, right where Halepohaku area is, in the alpine shrubland as you're going up the summit, and then up at the peak with the alpine stone desert. It is important to acknowledge that Mauna Kea is a sacred and special place with deep indigenous connections for community. In the back is a beautiful picture of the snow goddess Poliahu of Mauna Kea. I know Kamalani used this picture, but it's just so gorgeous, so I had to. So this summer, as a PIPES intern, I got to help with vegetation monitoring up on the summit, so doing those quarterly surveys around Halepohaku, as well as the native habitat restoration, just constantly learning and understanding the research field methods this summer. I got to help with a lot of outreach as well. They host a lot of community volunteer groups up on the summit, so if you're interested, there is volunteer forums, I think, starting in August. And just watching them with the community presentations and events and meetings they have throughout the season. I also got to help with annual invertebrate monitoring, so being in the field, making those observations of vegetation, depth to ash, depth to moisture, and weather, which I will get into later. But I also got to survey those native and non-native species up on the summit. This is where I'll do the trigger warning for spiders, so Wailea, close your eyes, and anybody else who is weird to insects, this is your one and only warning. It was really special. This summer I got to focus on the Hogna hawaiiensis. So this is the wolf spider from the family Lycosidae. It is endemic to the Hawaiian Islands, so found on Maui, Oahu, and the Hawaii Islands. It is a dominant ambush predator on the Mauna Kea summit that relies on resident species and aeolian debris to feed off and hunt. So it relies on those species that get blown up from lower elevations like Halepohaku up to the summit with those strong winds that the summit has. This spider is really, really important because it has a lot of ecological roles. Although it's so small, its impact is very, very large. It helps with a lot of nutrient cycling, the food sources, and it is an ecosystem regulator. So again, what we do know about this wolf spider is about the habitat on Mauna Kea that it does, prefer and has been found in the past. So that alpine stone desert and that large cinder area. And although we do know a lot of it, there is a lot that we don't know. So it is important to, the work that I was doing this summer, the research, to understand it more, because it is a really important factor in the ecosystem. So this summer, I analyzed 4 years of annual invertebrate monitoring data, just to help better understand how to find the wolf spider, just to find the best suitable habitats, where the Hogna, the wolf spider, is present or absent, and to protect those key habitat areas from future human impact. such as maybe some future development or human traffic along the roads. And as well to improve that survey methodology to focus on the wolf spider. So going into the Huaka'i, I did a lot of annual invertebrate monitoring. So these are the methods that we use. This is an area of the map. So on this lower kind of green spot is Halepohaku, and then going up the access road onto the peak of where all the peaks are at. Native species monitoring and early detection of those invertebrate threats. The elevation of the summit ranges from 9,200 feet at Halepohaku to that 13,803 feet peak. It's really tall. There is 111 total sites surveyed annually at 17 different locations focused on the cinder cones and in those highly trafficked areas. And this summer, we focused on the baited pitfall data, which specifies on those crawling insects to catch it in that trap. Now, what that trap looks like, this is our baited pitfall trap. Up on the top, we have the cap rock, which we bait with tuna. And on the inside, we have a wet wick to provide that moisture and water for the spider, as well as bait inside the cup, which is the tuna, to provide that food that they like. We also put in some rock in there for some habitat. And it's a dual cup. So it's 2 cups, and then we dig a hole down in the dirt, and then we put the cup in, and then put the rock on top. So very, very pretty simple, I think. So yeah, these traps are placed at every site for 3 to 4 days, and any unknown specimens that are caught in the trap are taken to the lab to later then be identified. These are some pictures of the wolf spider observed during trap pickup days. So on the left, it's on the little rock. It's really cute. And then here it's in the cup, so in our trap. And then here's Anu holding the spider. She's so brave. So during those field days, we investigated environmental factors associated with the presence of the wolf spider. So several habitat types were measured at each sampling location. These factors were focused on because they're best expected to influence habitat suitability and those conditions that I thought the Hogna might prefer. So I focused on these factors that are up on the top. So measuring how deep volcanic ash is from the surface at each trap location, how deep the moisture occurs in the soil, so how much you have to dig to find that wet soil, and then whether vegetation is present or absent at each site within 10 meters of the radius trap. So looking around, being like, do we see grass? Do we see native, non-native species around? As well as the substrate, so is it ash, cinder, or is it glacial bedrock ground that we're on? And the site, the geographic sampling location, and the year that the sample was collected. So in order to see if there's a relationship among those environmental factors and the wolf spider presence, I use multiple logistic regressions and use model selection using Akaika Information Criterion, AIC, to determine those best predicted factors. So we're gonna get into some graphs and some numbers, so just follow along with me. It's really interesting stuff, I promise. So the candidate models we used for our models was looking at ash and moisture, vegetation, moisture, substrate and year, and site and year. So why was it important to look at those specifically? I believe that throughout my summer experience, these factors can best explain the relationships and the patterns, as well as like population booms and busts. So the statistical methods that predict the probability of the species being present based on those environmental variables is important because then we can protect those areas from higher predicted wolf spider presence, again, from that human disturbance, that foot traffic, and further development to protect these spiders. So going into our ROC curve, this graph tells us which model predicts the data best. So the closer that the AUC is to 1, the best that the model will be. So here we can see that site year has the highest AUC of 0.763, so it's this purple line on the top, followed by substrate year and ash and moisture. So I chose to focus on the site and year and the ash to moisture. So is this model perfect? Is this the best model we can use? Probably not, but it does help us determine which model provides the best explanation of the data while avoiding models that are more complicated than necessary. So it can't explain all the variation, and there are additional variables also influencing wolf spider presence. And during running those statistics, the triggers and errors did come up. So it's not perfect, but some of those errors could be due to a smaller data set, bias data that we have or environmental variables that just weren't measured, as well as maybe some weather gaps. But it's still significant and it's still really important, and this data we can still make a lot of inferences from. So going into my first model, looking at ash and moisture. So asking the question, do local habitat conditions influence wolf spider presence? Looking at ash, for 1 unit increase in depth to ash, the odds of Hogna presence are estimated to decrease by 4.2. However, the depth to ash was not significantly associated with Hogna Hawaiian presence. Looking at the moisture, for every 1 unit increase in depth to moisture, the odds of Hogna presence decreased by 6 to 7. So this tells us that depth to moisture has a significant negative association with Hogna hawaiiensis presence. So the more moisture that is involved, the less likely the wolf spider is to be in that area. So yeah, here we're looking at the measure how the odds of Hogna presence changes as those predictor factors increase. And then looking at the OR, if it's 1, it has no effect. If it's less than 1, the odds increase, and if it's greater than 1, the odds decrease. This is the graph, is a plot showing predicted probability of Hogna presence by depth to moisture, where 0 is absence and 1 is presence of the Hogna. So the blue line right here shows the negative relationship between moisture depth and wolf spider presence. So it can influence Hogna habitat because moisture does affect. So looking at the soil conditions and the importance of like the burrowing habitat that the wolf spider prefers, it's in drier, larger areas. So again, as moisture increases, conditions become less favorable for that active ground hunting spider that occupies those dry volcanic substrate areas. Going into my second model, we're looking at the model predicted probability of Hogna presence by site location and collection year. So here we're looking at the sites down here and the years in different colors. So we're asking the question here, do spatial and temporal differences explain Hogna presence? So looking at the graph, the sites consistently had higher predicted probability than others, so showing that important habitat difference, as well as the sites with higher predicted probability of presence are located around 11,000 to 12,000 feet in elevation. So these 4 locations on the right have constantly had higher probability of presence of the wolf spider. So those differences among the years reflecting changes in environmental conditions. So maybe in 1 year there was a drought, heavy rain, temperature, and climatic variation. So the different sites, it's important to, look at that elevation, habitat type, vegetation, temperature, substrate, and prey availability. And in the year, reflecting that rainfall, snowfall, drought, seasonal weather, what has been going on that year. So yeah, really cool inferences over on the right. And then these areas, since they are on lower elevations, this is what that area kind of looks like. So this is VLBA North. This is where one of the pu'us where they had higher chances of finding the Hogna. And then this is the upper South Lake Cinder Cone. So these lower pu'us over here in this area is where they're more likely to be found. So yeah, they're up in high elevation, but they're lower than most of the pu'us. So it's in that like lower area where they have been found. Going into my ho'ina, sorry, Wailea. What we can conclude, so together, the results suggest that Hogna hawaiiensis presence is influenced by both local habitat conditions and broader spatial and temporal variation. So with the ash and moisture, the moisture was significantly associated with Hogna presence. So those local habitat conditions do influence where the spiders occur. And site and year, the highest predictive performance was the most effective model for distinguishing Hogna presence in that area. So the best model really does depend on what you're measuring. So if you want to measure different factors, then you can go ahead and play with those. So going into the future of this research and CMS, what can maybe be improved? Maybe leaving the traps out for a longer time span. There was a study done by Howarth where he said that they left the traps out for 1 to 2 weeks. Maybe it was 1 week. And they found more wolf spider presence since those traps were left out for longer. And as well as maybe we can survey throughout the whole year instead of just the summer season. Adjusting those trap methodology for wolf spiders, since that trap, that baited pitfall, was mainly made for the VQ bug. And then continuing studying those relationships and the role in the environment and how important of a factor that the wolf spider plays. So interpreting to the community, tying this all back to the community, The wolf spider is an important part of our ecosystem that is unfortunately understudied. So doing the work to help us better understand its role allows the community to connect to the wolf spider and the place that they call home. This summer I found new perspectives on community and ecology, being able to connect with the Aina, which are the elders and the teachers that help us feel more connected to place, and to continue learning and caring for those spaces. Going into my ha'ina, these are some cute pictures throughout the season. And yeah, this internship confirmed that I want to continue working in conservation. It brings me, that ecological research, that indigenous knowledge and community. So wherever my career takes me, I want to slow down, listen first, and approach conservation with intention and humility, and continue working for my community back home, whether that's in San Diego, Yosemite, or Mexico. One of the greatest transformations I experienced throughout this internship was learning to slow down. Before coming to Hawaii, I often approached challenges by wanting to act immediately and finding a solution right away, but during all the time I spent up on Mauna Kea, I learned that meaningful conservation begins with observing, listening, and intentionality. I became more connected to the Aina because I learned that conservation is not about rushing to fix something, but about understanding that relationship that already exists. So thank you very much. Thank you to the Center for Mauna Kea Stewardship for welcoming me with open arms and teaching me all the ropes of how to be the most successful intern that I could be. Thank you to my mentors, Anu, Taylor, Kaya, Justin, Kaleo, for just showing up every day and just being the best. You guys taught me so much, and you guys inspired me so much, and I hope that one day I can be amazing ecologists and protectors of the Aina like you all are. And thank you to PIPES for allowing me to be here in this beautiful place this summer. It has been such a privilege to be here and stand up here and talk to you. And I could not be more grateful. This summer has genuinely changed the trajectory of my life. And thank you to my family back home who are watching. Muchas gracias, mami. I could not have done it without your support and everything, every single day when I didn't want to get up. And thank you to the University of Hawaii at Hilo for housing us. Those dorms are amazing. And thank you to our sponsors, and thank you very much. Emi Ninao. Yes.
Hold on, comma.
Hi, Amanda. That was an amazing presentation. Mahalo for sharing.
Thank you.
My need now to you is, since you've been in the Aina this summer, and the Aina has shown you a lot of things, and you built that connection, what has the Aina taught you? Like, what is one of the most valuable lessons that has taught you that you're going to bring back home?
That is a great question. I feel like one of the biggest things that I want to take back home is learning to slow down. I feel like back home in California, it's such a hustle culture. Like, people are trying to get to the next thing right away and do the next thing and be bigger or be better. But I feel like being here, it's helped me appreciate that the trees don't grow in a day. The trees grow years and years. They go so slow, and I want to be able to move slower and with more intention throughout my life. I feel like we always focus on the next thing, and I want to be able to focus on the now, just sit here and enjoy everything that's in front of me and just slow down. And I want to be able to take that back home with me and follow through, slow down with my days, be more intentional, and be grateful for everything that I have and everything that the Aina does provide, One of my favorite things that I've heard is, use what get? Use what get. So I want to be able to use what I have available to me and not depend too much on external factors and external people and companies. So hopefully I can take that back home. Yeah, thank you. That was a great question.
Mahalo.
Amazing job, Amanda. I also had one question. I was wondering, of all your days up on the Mauna, was there any one particular highlight that you could share with us? One day that like stood out from all the others?
I feel like I've already talked about this day so much, but it was my first week, my first or second week, and we were setting out traps up on the pu'us. I think it was in VLBA North or South, and me and Anu went up to one of the pu'us, and I talked to you about this, Kiana, during the site visit, but that day was just so special. It was a pretty hard walk up the pu'u. Is you just stick your foot in, and it's like, till you get to the top. And once we got to the top, it was just this like heavy feeling on my chest. Like you can feel the energy up there. And I had asked Anu, I was like, dang, like I can really feel the energy up here. And she had told me that that pu'u was part of one of the trails that the ancestors took way, way, way back to get to the Ats quarry that was like right across. I don't know if I'm saying this right, but it was a very heavily populated area, and there was like burials around us. So just feeling that energy that, you know, they talk about on their website, and they talk about the first week, it's like a very sacred, special place, but it felt so special to feel that. And I got really emotional. I cried. It was like this whole thing. The view was just beautiful. And it was one of those moments where I was like, I made it. You know, I didn't just make it up to the pu'u, but I made it here. I'm like taking that foot into conservation as like the first person in my family to go to college. So it was a really great moment, and I'm gonna take that with me, because it's just being able to sit like with your achievements and being able to also take in like, you know, an experience and feel the energy that a place can bring you. So many places on this island have that energy, but like just being up there was just crazy, and it's a privilege. People don't get to go up on those pu'us, and so being able to have that, you know, awareness of, you know, that space. So, yeah.
Mahalo nui for sharing. Can we get one more round of applause for Amanda? Up next, we have Mr. Kanoa Ponokipapa with the Indigenous Cropping Systems Lab, telling us all about roots to research, or research to roots, and the interconnectedness between it all.
Hello? Hello? Hi, everyone. Good morning. Isn't it a beautiful day? A glorious day. Well, I'm going to be talking about my adventure over this summer and all the things I learned. So the title of my presentation is From Research to Roots, and connecting academic agroforestry to practical community stewardship. And I'll be showing you why. But first, we're going to start with where I come from. So, the most important thing on a journey to me is the foundation. It's always getting started. What am I going to pack? What am I going to do? What's starting me on this quest? For me, that's my family. They've defined that. They've told me, or they've given me the opportunity to have so many responsibilities that I get to pursue. And they've educated me and put me in places that I will never be able to fully thank them for. Then, after like going through the journey and seeing my family as the foundation, I would say the next most important thing to me was my first ever mentor, which was Nick Francisco. He taught me the very importance of each and every time I pick up a plant and I know its name. Each time I use any kind of crop, plant, and just try and want to know the background for that kind of crop. So this summer, and then I'm a student at the University of Hawaiʻi, trying to pursue a degree in business with a concentration in agriculture. And through that, I found this really cool internship. I don't know if you guys heard of it, but I think it's called like PIPES or something. And then I signed up for it, took a long shot. I was like, can I do this? Will it work out? Then I got to meet with this really cool person, Ho'oulu. She's over there in the crowd. And we got to have our first talk, and it was like, I feel like it was like 2.5 hours, like our first, it was 2.5 hours, and she was explaining to me why each and every plant inside of germplasm is important. I was like, I love this person. Let's make sure we work together. So with that said, I kind of got to look into the missions of the organization that I was working for. So eventually I got hired by her, and doing an internship under her with the University of Manoa, their extension center, their college, CTAR, which basically has the mission of just teaching people about agriculture and using it for practical use. And then I got to join this lab, the Indigenous Cropping Lab. Sema's there, Ku's there. But we got to talk about, or it kind of just, the way that Noah Lincoln, person that described everything to us, was that we just want to get everyone to understand the adequate use of indigenous crops and indigenous cropping systems. Because what's better than doing what has been taught over and over and over again and using it in our community? So my whole entire goal this summer, it was to effectively make outreach material for the lab and try and talk to people, get connected with the community, and kind of create like the preliminary research, like being like a conduit for the research that they have and converting that into something that the community can use. Because like, what is research if it can't be used? The main people I worked with this summer, other than Sema and Ku, was Jiten. I got to meet her, I got to understand, and we got to talk, build Polina, and then I also got to meet Aina. She was a past PIPES intern, and we got to talk about all of our goals over the summer. The biggest thing to my mentor was giving us quests, and we were gonna have to coordinate how everything is gonna work out in the future. So, starting with that, we had weekly meetings. We met every Monday. We kind of got to know each other really well. First week was almost completely Polina. We wanted to understand each other's strengths, understand each other's schedules, understand where everyone was gonna be this summer. So then we could formulate a plan for what we wanted to do. And then this was like our objectives of what we were gonna complete over this summer. We found that creating videos and using videos is such a good way to share information. We all use Instagram, maybe Snapchat, whatever you use to watch videos, but it always conveys the thought, you know? We wanted to make something that isn't AI generated. It was using, creating content for like the media that was information from the lab. And there was so much aspects to that. We had to learn how to kind of like script together, edit videos, create videos, produce videos. As you can see, that's what we're doing in that scene. And then one of the other important things that we learned to do, kind of basing off of that, is interviews. We got to learn how to schedule meetings with people, coordinate certain things that we want, and then we thought it was important that the community gets to have a say in what is important to them. So interviews are probably like the best way of doing that, because you get to hear right from their mouths why it's important. And then we thought posters were really important because, again, like we all see those posters all over the place, but it helps give information to people and share subtle things that you might not know, or you might know, or can just be added on to. Easy ways to share information. And then the final objective that we had of this, or my main objective, was learning how to label a germplasm. A germplasm is just basically a fancy way of saying a living seed bank. It's nothing more important than that, but it's just preserving varieties and, or species and varieties. So like we're saying, because we want to make sure everyone understands the importance of leading into the next topic. Oh. Where is it? Well, I had a slide presentation that said agroforestry and its definition and understanding it, but I don't exactly know where it is. So I'm gonna go off from the top of my head. I'm gonna give you guys the rundown. It's basically like a cake. Think of agriculture as a cake. We don't want just one slice of vanilla. We want everything. We want to add those chocolate chips, that whipped cream, everything that makes that thing look good. So basically, making agroforestry is adding or replicating kind of like the effects of a forest, the natural shading, the natural lighting, windbreaks, to help with reduce erosion, like all of those things. And then using that and understanding its importance in agriculture today, because we want to make sure that crops, all crops, get a say in the new world. So going on to my huaka'i, I got to, over the summer I learned a lot about like ava. That was my main thing. I was doing, for that part, for like the videos, I did mainly the background research and the acting, which is the only one I was kind of good at. But I got to learn so much about ava and the appreciations. There's so many people I appreciate for reading through the research and then meeting at the conference we got to see. And then finally being able to like even harvest one. So this is us harvesting ava nene. And then kind of like the defining things over the summer and then learning what we were doing. We got to do like field work, which was really important to learning outreach, because now we get to integrate what we want other people to do. Like learning the importance of adding nematodes and just being able to be in person, be present, and learn about each thing. Next thing, oh gosh. Wait, do you guys mind if I, like, this is like an older presentation of what I had, so it's like, oh, that's awkward. Just basically, the other thing we got to do, I got to do interviews over the summer. We had 3 different interviews, or we had 5 different interviews. One with a big role model to me, Ed Johnson. He got to define how important ava is to me and how important it was to him, and going through and protecting different varieties. And then for DHHL projects, which also told me the importance of each and every one of their places and the ainas around. We got to do scripting and acting, like I said. This future sense, but we have videos that we're gonna produce that were the ava palalas, and we're teaching everyone about the importance of ava in each variety. And then the other last thing was outreach. It was really important to be able to communicate with the community, actually have that connection, see how to teach a child what to actually do when you're in a situation, because that can be so much more different when you're reading or doing research and then applying it. You're like, what? You don't actually know what that is, or you don't know how to, you're scared to touch dirt, or, you know, there's so many different things. So outreach was very important because it was our kind of way to bridge our way through the community. And then this was our DHHL project. My mentor and our lab, they worked on something called the Hawaiian Homelands Community Forest and Family Trees Project. It was basically putting out trees into the community and into people's hands that will be helpful. So we got to go all around. We got to go, I didn't get to go to this meeting, but we got to go to King's Landing. They have a nonprofit down there, and their emphasis is also on perpetuating agriculture. And then we got to go to the Keaukaha Panaewa. It's right across Home Depot, and it's like really cool there. They have so many different trees, but we got to learn why they started and what is actually important to each of these people in the community. They are the representation of the community. So through this project, we got to learn about all the people who get like plants and stuff from them and from the lab. Then we also worked with Panaewa Hawaiian Homelands Community Association. It's right across, it's like right behind downtown Longs, or uptown Longs. And it shows like, I don't know, it's kind of in like the starting process, but they have where they want to clear out that forest and bring back native species, lay-making species, mainly of emphasis on llama, because it's one of the oldest and like most iconic hardwoods. And then we also worked with Maku. We got to learn all about what their commitment to and how they're shaping the community there. Oh gosh. Okay, well, I also wrote subtle descriptions on each of these, so I'm gonna do them by memory. The first one's supposed to be Nanea. It's basically, shout out Sema, a cover crop that helps fix nitrogen in the soil, hold erosion, and is really good biomass. Ko, there's many stories. One from, I think Tonga, talking about how it emerged from her feet or head, along with Ava. And I think is the sugar king of the Pacific. And then long gone, this is there, but it's mainly there to become mulch in the future, so don't really have to look at that. And then Ava, my favorite, the one I got to learn mostly about, but it's can be served as a drink and has euphoric effects alongside it. Ulu, it's like the king of starch. Don't get mad at me, whoever likes kalo, but I feel like Ulu produces so much food, and it's like the perfect thing. Plant it in the ground, take care of it for 5 years, and it's gonna take care of you for a lifetime. And then Niu. Someone likes to call this the nectar of the Pacific. It's the very embodiment of the Pacific Island community, moving from island to island, being able to survive there, and used as almost a base in a lot of dishes. And then la'i, used for lei, medicinal purposes, and even cooking sometimes. Just tea leaf. I got to map out, this is our germplasm. This is our demonstration lot that they have. We wanted to focus on like each individual small plant that is there, so we can bring people around, demonstrate how an agroforestry system can actually function. So it has a mix of wide varieties. I'm guessing gotta be over 200 different varieties of species. Within the different plants I talked about, it has sugarcane. You can see each of them labeled right over here. They have, I think it's 34, but 24 maybe. But each one having its own story and its own background. And that's like the main thing that we wanted to focus on with this demonstration lot, is showing that we can plant all of these things together and they'll all produce functionally. And not only that each of these species are like important by themselves, but we wanted to make sure that varieties are also shown in the spotlight, because varieties are also important. They have so much history, especially for plants that are like somatic mutations. It took time to grow to that to that plant. And we want to emphasize that. Make sure that you care about the name of your plants. Not only, oh, it's a coconut, but it's a certain kind. Or not only is I'm going to serve you a bowl of ava, but I'm going to serve you a certain kind. Because those each carry significance. It's reclaiming that name. It's understanding the history behind each and every one of those. We have the breadfruit species that are here. And then we have Nanea, where it's all plotted out. And then ice cream bean. And then we have ava, again, the main one I focused on. If you guys want to know anything about each and every one of them, I took it upon myself to learn about each and every one of them. And I had posters that I had made over here, but I would just kind of go off the top of my head. We got to learn about Hanakapi'ai, which was biasly one of my favorite. It had a different color name. It's like uli uli. No, I'm not even gonna try. But it just means crazy mixed up colors, because it gives you lucid dreams. And just learning that and learning the effects of what each of those can do was so like encouraging to me. And it's really what's pushing me forward into my hainan. As a person I am, I hope that my research and what I could do, creating annotative bibs, scripting, creating posters, creating these interviews will allow for people in the future to understand the same importance I got to understand through this experiment. My main thing that I want to see is I want to see people drinking ava and being able to be immersified inside of the different plants that are there that we all can use instead of being reliant on top of things that come from so far away. And then, and I also want food sovereignty, and also just like, I want people to know the importance of the plants and plant them themselves, because that's important. And I hope my posters will be able to perpetuate that within the community. And then through this entire experiment, the main thing I wanted to learn was organization for myself. And going in every day and being able to go through each experience and show up every Monday to 10 o'clock to all of our meetings, and then be able to plan out what we're gonna do that week and structurally make out a plan for tackling all of these goals has really helped me with organization and understanding teamwork. So much personal development on my part. I want to give a huge thanks to my mentor, Ho'olu Erica over there, Akaka. She's like the perfect person for understanding everything from personal problems to what I got to go through this summer. And she's honestly helped me. She hooked me at the first 3 hours we talked. I feel Noah Lincoln has helped me so much with helping with research and connecting me with people throughout the community. So thank you so much. Tommy, he's been like one of our registered adults at the lab, because it gets kind of hectic. But thank you for dealing with us over the summer. And Dolly, we got to inject nematodes inside of ulu trees with her, which was so fun. Never tried it. Just like close your eyes when it happens. You can ask Ku. It gets all in there. Sama, been a huge help for me. He did pipes last year, so like every week I'm asking him the same questions. I'm like, what do we have to do this week? What assignments is going on? So thank you so much, Sama. Thanks for dealing with me. Jacob, our other registered adult, he's helped us so much with like going through and communicating with other people. He's also helped me with like all of my writing assignments. He's very on it. And then Ku, most importantly, my biggest op in the lab, but helps me push to be better than her, because like that's the whole point of having competition. So thank you, Ku. And then I want to say a big thank you to all of you guys who are here, all of you guys who've put together everything that I've got to do this summer and inviting me along this journey. It's been so much of a personal impact for me. Learning about how important, not how important conservation is, but how important I can be within conservation. Being a gear to turn our society forward, that's cool as hell. And then, thank you to all the other sponsors, the Indigenous Cropping Labs, awesome. Systems Lab, awesome. Kipuka, thank you so much. And yeah, to conclude, thank you all for listening.
Amazing job, Pono. We have time for one question. Do we have one question from the audience, online or in person?
Hi, Pono. How did you enjoy doing outreach work? Is it something new to you, and is it something you want to continue to do in the future?
So I think working with the community is really important. I've done it from a really young age, luckily with my family, with one of my aunties being a coordinator for events and stuff. She's put me in community settings, and it's developed me so to a way where I understand work ethic, where I understand the impact of everything I do, where I understand at the end of the day, it doesn't matter how much it hurts, but how much you've put into the aina. And I think being able to put myself in a position where I can make other people feel that way, especially younger boys that were kind of like me, to be able to put them in a position where they understand that they have role models out there and they can be a positive impact within our community is the main reason why I like doing outreach. And seeing that, I think I went over this, but seeing that glimpse of curiosity within a child's eye, it's the very reason why I like outreach, even in adult size, sometimes.
One final round of applause for Kanoa Pono Kipapa. Up next, we will have Miss Mina with the Ilala Biodiversity Labs telling us about Palila on the Mauna.
Hello, everybody. Aloha, mai kako. My name is Mina Adaba, and I'm really excited to tell you what I've been up to this summer. Okay, I had the honor of being mentored by Hannah and Sam and Dr. Natalie Graham in the Ilala Lab at Hilo. Shout out to them. They're on Molokai doing CRB work right now. So, our thoughts with them. Hopefully there's none. Okay, so I grew up in Minneapolis, Minnesota, and being part of diaspora and then moving and burning it up in the Bay for all of college, people often ask me, what the heck is Minnesota? What is it? Is it Canada? Is it on the East Coast? Is it in the continent? People do not know what it is, but I don't blame you. That's okay. This is not America too. So anyway, but I really was raised by the Aina. Minnesota has almost 11,000 lakes. So I grew up in the water. You guys probably think it's kind of gross water because it's warm and fresh water, but I love it. And my school had a farm, and that really established this love for working with my hands. I remember picking worms out of the dirt and handing them to my grandmother, and she's like, I don't want this. But I was like, oh, this is important. This is an important being. Anyway, so this is me. I was obsessed with tomatoes. I'd eat them like apples. Oh, yeah, this is us in our garden. That's my dad. Oh, that's a tomato plant. And then the winters. Minnesota has the most frigid winters ever. Sometimes it gets to -40. But the thing is, Minnesota also has the kindest people. And I remember, like, I have all these core memories of big blizzards and, like, driving home from school. And, like, literally every single block, there'll be multiple people with, like, out of their cars pushing each other through the snow. And, like, you don't have to say a word to them. Like, they just, like, come out, they help you, and then they leave. So, especially now, when we're being attacked by the federal government, like, they can try, but, like, we're so strong. Anyway, so that's me with the frog. And then, yeah, just growing up, continuing to be in the world of conservation and research. I've had the privilege to work with a lot of different communities around the world to continue to build that pilino with the Aina. This is my family. And then this photo is really special to me because it's the first time we saw the Pacific Ocean with my Korean side of the family since they were forced out of the country. So I'm the proud granddaughter of refugees on both sides and the daughter of immigrants. So I carry them wherever I go. And then this is the other place I call home, Turkey. Okay. So coming to PIPES, one of my primary goals was to understand how can I be in this world of research in a way that doesn't reproduce these colonial patterns, because otherwise I don't want to participate in it. You know, it's very easy to be reinforcing the harm that colonization has had on places all around the world, and especially here. So yeah, my project was focused on the Palila. I don't know if you guys, I think most of you definitely know what the Palila is, but it is an endemic honey creeper on Hawai'i Island, and it lives on Mauna Kea in the Mamane and Nayo forests. And it's the last of 16 finch-billed honey creeper species across the Pai'aina. Okay, so the Palila, they eat 3 primary categories. One is the Cydia moth larva. We talked a little bit about this on our huaka'i, but this larva lives and grows within the pods of the mamane tree. And then their primary diet is the hua and vahiano ano mamane, which are the seeds in the pods. And then they like supplement with other parts of the forest as well. So why Palila? Why are we focused on Palila? Well, Palila for you know, as long as people have been on Hawaii, have been very culturally significant. This is a mele that was written for Queen Emma when she was ascending Mauna Kea. It says, "Ika leo honeo ka Palila, oia manu noho kuahivi." The sweet voice of the Palila, those birds that dwell upon the mountain. And then now today, people really understand the importance of the Palila, not only culturally, but also ecologically for the forest ecosystems. You guys have probably seen the mural in downtown Hilo. Yeah, it's beautiful. Okay, another reason that we're so focused on the Palila is because of how much their habitat has decreased and the impact that this has had on the species and population numbers. So there are only about 666 individuals, like an estimate, on Hawai'i Island and everywhere because they're endemic here. So the red is their historical range, and then the yellow is where they currently reside. Yeah, so the population has dropped 90% from 1998 to 2024. But there are a lot of groups that are working to restore these population numbers and help the forest regenerate with the help of the Palila, because they have like a very reciprocal relationship with the mamane trees. So they are like a big way that the trees, that the seeds end up spreading around the forest. Okay, so the way that we studied the health of Palila we looked at the levels of Lepidoptera, so like the moth species, or like moths in general, and how this parasitoid wasp impacts the moth species. Because the moths are really important for the Palila diet. That's why they're protein maxing. But the parasitic wasp is a potential competitor for them because they also prey on the larva. So this is a project that's led by Noah Hunt and his team at the DLNR, and then also the Elala Lab. Yeah, so we were looking at our Mamane forest habitats on Mauna Kea equipped to support the arthropod, so like the insect and spider, but mostly insect, diet of future Palila populations, and then what is that relationship between this protein source and the potential competitor? And then have the arthropod communities on these sites changed over the past 2 decades? Oh, this is a photo from Reina. Yeah, this is the Mamane, but you all planted that. Okay, yeah, this is where I worked on Mauna Kea. Okay, so how do we do this? We start by collecting insects. This is done using malaise trapping, which is what what this huge tent thing is. So after collecting insects and doing field work, we brought the samples back to the lab, and I spent a lot of the summer sorting insects and counting them. These are, bug warning, but yeah, I think they look pretty cool, so I take it back. Okay, so these are the hymenopterans. These aren't parasitoid wasps, but they are other species of wasp. So essentially I sorted them to order and then counted the number of individuals, which is totaled here. Yeah, I didn't know until like the last day that I didn't have to count each springtail. So I was like, I was frustrated. Not by that, by the springtails. Okay, so how do we actually use DNA to identify species? This is with something called DNA metabarcoding. Bear with me if you have experience, but this is the thing with DNA work, is that I think a lot of people feel excluded from the world and from the technology because there's so many dumb ways that people have like created it in an exclusive and exclusionary way. But every single one of you can do this. Like it is just something that you have to learn. So yeah, with that, basically the first step is to extract the DNA from the samples, from those insects that we got. And then we do something called TrueSeq PCR. So basically we're cutting a piece of the DNA that, like a specific section that we want to look at, and then replicating it over and over and over, so we just like have a bunch of it. And then we run a gel electrophoresis, so like we're just checking, did everything go okay? Like, do we have enough of that specific part of the DNA? And then we do something called indexing PCR, so we're attaching like labels to them so that we're able to trace them back to what sample they came from. We run another gel. And so basically you do that with every sample that you have, and then you combine them all into something called Dirty Library Pool, which they should rename. And then you use a tape station machine and a bead cleanup to basically like ensure that there are like not that many contaminants in your samples, and then we send it away. And then it gets sequenced, and we take the DNA sequence that we get back and we compare it to databases of known sequences. Okay, so this is like the extraction process. That's me quantifying DNA. This is called a nanodrop. It takes 1 microliter of your solution, and then it'll tell you DNA, RNA concentration, and then protein and salt contamination. So pretty cool tech. But again, I didn't know any of this before I came. So thank you, Hannah. This is me performing the PCR. And this is like within 2 projects. So the first one was with the Palila, looking at the arthropod communities. And then the second one was in taking samples that communities send of potential coconut rhinoceros beetle, which is, I'm sure a lot of you know, is like a very harmful invasive species here, to test if it indeed is CRB or not. yes, this is the gel that I was talking about. It's just like a, so, okay. This was the fieldwork that we did for CRB. It's focused on eDNA. Unfortunately, CRB was detected for the first time while we were in the lab, or on the Hilo side. But this is in Kona. We were paint rolling trees to collect DNA that was on the tree, and then we extract that DNA, and then we look if CRB is present. Because like the thing with CRB is once you can detect it, when a tree looks like this, with this marking on it, the tree is very much like, it's really hard to come back from that. So eDNA is a really exciting way of potentially doing early detection methods and also really precise detection methods. So that's something I want to continue working on. These are the CRB. Boo. Okay. All right. So when I was running that gel, this is basically what you get back. The middle here is just a solution with DNA of a specific length, or like it's called the DNA ladder. So you basically like use it to scale the samples that you get. So each well is one sample, and then the dark marks, if they align with this dark mark, that means basically that the PCR was successful, and the primer we used was like applicable. So yeah, we ran a couple of those. And then we're waiting on the data to be sequenced and to come back, but in looking at like, because I sorted through all those and counted all those insects, I used that data to look at potential trends in arthropods on Mauna Kea, and it's shown here. I would wait for the actual sequencing to come back to like fully analyze. But yeah, this is like mean abundance of individuals by order, and then across the collection dates from November 2025 to May 2026. And then if we're looking at Hymenoptera and Lepidoptera, specifically by site, this can be used to inform potential conservation methods, because it is important to understand like what is the abundance of this Lepidoptera at these different parts of the forest, and how are the wasps that are in that area potentially affecting them. This is the DNA sequence that you can get. This is a CRB, like known DNA sequence, but this is like what gets returned to you, and then you put it through software to compare with like a full database. So you can see here, it is like a 100% match with the known genome. So yeah, that's coming back soon, hopefully. Yeah, so, man, have I grown? Yes. The DLNR work that Noah Hunt is leading is ongoing, and there's a whole crew of really passionate like land stewards that are working with him. And yeah, so the research that I was doing in understanding the communities that are present is the basis for implementing different experiments that will, like in hopes of increasing the amount of that Cydia moth within the forest habitats of the Palila. And then eDNA, I think that's what I feel really passionate about, because I think it's like a much more responsible and like less obtrusive way of doing research, especially INA-based research. And I'm excited to teach other people about it, because it sounds very technical, but honestly it's something that can be made very accessible. Just some people refuse to do that. Yeah, so I'm graduating from UC Berkeley in the spring with a BS in molecular environmental bio and concentration in global change ecology, and then a minor in human rights. And I've had the privilege of working in a lot of environmental justice, advocacy, decolonial research, like worlds, and having a whole bunch of incredible mentors. And a lot of those that work has been centered on coastal communities, particularly for Pacifica nations. And I hope to continue to build this pilina with people and place wherever I can be helpful. Yeah, so my experience with PIPES has really expanded where I see myself in the world of research and in Aina stewardship. I'm really excited to continue all of our relationships because we're from all over the world, honestly. And our family, we're from diaspora from all over the world as well, but we're all united in our kuleana of Malama Aina. And there's a Turkish proverb. I've been teaching Kaysen my Turkish proverbs, but this is damla damla göl olur, so that means drop by drop a lake is formed. And I think that that's really a testament to the world of conservation and Aina work. Yeah. Oh, I also like was sketching in the field. So that's like, I don't know, that's like a way that I feel connected to the work that I'm doing and the place, because you become very in tune with the intricate details around you. Yes. Okay, mahalo nui loa to Hannah and to Kayleni, thanks for coming, and to Dr. Graham and Sam for your mentorship. This has really changed like my expectations for a workplace, honestly, because of all the support and like steadfastness that you guys have shown me. And then thank you, Noah Hunt, Elizabeth Lowe, and Kylie for letting me join you in the lab. And also to Kiana Vai, Lenalu Hinano, and Revlin. PIPES has been an incredible place to be this summer, and I'm so grateful for the network that you create. Oh, and also, and Sharon. Yeah, and thank you to the NSF-RU. Appreciate you. Aileme teşekkürler. Sizin sayenizde buradayım. I'm here because of my family, and I carry you wherever I go. Mahalo.
Amazing, amazing work, Mina. Do we have any questions for her? For Mr. Sema.
Mahalo, Mina. Wonderful presentation. How would you like demystify eDNA and like qPCR? Because I mean, like for me, growing up in school, we never learned about these techniques. It wasn't until we got to college. So like, how would you communicate that to younger people who are interested in conservation or molecular work? Thank you.
Fantastic question, Sema. Thank you. Okay, I think honestly, step number one is at least explain the acronyms, because people do not people are just throwing around jargon, and I think some of it is intentional to be exclusive in the world of academia in a completely unnecessary way that blocks out people that do not have the opportunity that others do, and those are the individuals that should be doing the research because they're the most informed in other aspects. So I think patience and different ways of learning are really key in this. And also, yeah, I think that there are ways that molecular work can be taught even at a younger age, just to establish that understanding of the data that we cannot see with our eyes. Yeah, I had something else, I forgot.
Can we get one last round of applause for Mina? And then staying up on Mauna Kea, up next we have Mr. Kaysen, kind of giving us another aspect to the work that's happening up in those high elevations.
Aloha mai kākou. My name is Kason Guillermo. My project was on koa outplanting health based on dibble size and shade shelter type. So who am I? I was born and raised in Hilo. I'm in the Mo'aina pathway with PIPES. And I think a lot of my conservation motivation comes from when I went to Ka'o in middle school. And at Ka'o, they do a lot of like place-based learning. So the whole year, you'd go to one place and you learn everything about that place, whether that be Honokea or Haleolono in Keaukaha, or that could be down at Lihikai at Onekaukaha, working with the kalo, or even up at Keho'oka'u, learning about the water cycle. Right now, I go to UH Hilo. I'm in the biology, conservation, ecology, biology pathway. And another motivation I have for conservation, I'm calling it like ancestral FOMO, where it's basically like, I hate the feeling of like missing out on things that like people before me could have experienced. So like hearing about how in Hilo, the break wall, pre-break wall, you could like surf, you could gather limu, but now it's like the swell doesn't come in, it's all brown at Hilo Bay. Or how there's so many like yellow birds in the forest that Kamehameha could have a whole yellow au'ula. And yeah, that's like, I hate that feeling of like missing out on that kind of stuff. So this summer, I'm working with Mauna Kea Watershed Alliance, or MIKWA, and they focus mainly on managing the Waipahoehoe Management Unit, or Waimau, and the Kanakaleo Nui Bird Corridor, or the KBC. These sites are located on the eastern slopes of Mauna Kea, from 6,500 to 8,000 feet. And at these sites, MIKWA does outplanting, furrow ungulate control, and fence work. They also work with other organizations up there like the Department of Forestry and Wildlife, Department of Hawaiian Homelands, and the Hakalau Refuge. So my project mainly focused in on the KBC. And the purpose of the KBC is to extend the low elevation forest to the higher Mamane elevation forest, to get out of the range of mosquitoes, which are a vector for malaria, or avian malaria. And the KBC is 515 acres, and is mainly planted with koa in this lower area. And we call this this is the boot. It's kind of shaped like a shoe. And most of my planting was done in this bottom foot area. And the Kanakaleo Nui Bird Corridor is named after Pu'u Kanakaleo Nui. And it's named after a guy that used to live up there during the time of Umialiloa. And his name was Kanakaleo Nui, which means the man with the loud voice, or the loud voiced man. And his role up there was Umialiloa would tell him orders, and because of his loud voice, he was able to deliver that message to all the other parts of the island. So for my data collection, I surveyed 300 koa that were planted in April. And they all had 4 different combinations of dibble size and shelter type. And they were labeled as D25 plus shade cloth, D25 plus yellow plastic, or SC10 plus yellow plastic plus, or SC10 plus shade cloth. The soil in all these plots were stony loam soil, and all the plots had this kind of like gulchy, hilly terrain. I surveyed them in early July, and all these plants are watered weekly, but they were not watered if the Mauna provided sufficient rain. So I was told I need to define what a dibble was. So it's this yellow pot thing. It's kind of like an ice cream cone. And D25 means it's 25 cubic inches of volume, and SC10 is 10 inches of cubic volume. And then on the left, this is the yellow plastic shade shelter. And on the right is the shade cloth shade shelter. And for the shade cloth shade shelter, all of these posts are set up on the east side of the plant to protect it from the harsh morning sun. And both shade shelters are just to help the koa in its early stages of life. So originally, my plan for research was to base it on how many plants are still alive or dead after planting. But fortunately, most of the plants are alive, but then that meant I had to change how I'd be judging the koa and which factors are the most efficient. So I set it up into healthy, moderate, and poor. These are examples of healthy koa. So it's all green leaves. The leaves are turgid, which means that the leaves are still firm and they're full of water. This would be an example of moderate health koa. So I kind of judged it based on half of the leaves are like damaged in some way. So this purple reddish color is evidence of like frost. And then this one, it's missing leaves all down here. And then the leaves are kind of yellowing. This would be an example of a poor health koa. So all the leaves are wilting, everything is yellowing. Even these fresh sprouting leaves are wilting, and that usually means that it's like on its way to dying. So after getting all my data, I did a chi-square test, and it showed that higher proportions of the D25 dibble koa were found to be healthy rather than the SC10s, and higher proportions of the shade cloth koa were found to be healthy than the yellow plastic shade shelter. So outside of data collection, I mainly did data collection for one day, and the other experiences I had on the Mauna, we're collecting seeds. This is like off of Mauna Road. And we're collecting more koa because we already have a bunch of the other main plants that we plant, like mamane and aveoveo. So we're trying to diversify our personal seed bank. And then, if you guys didn't know, we have like feral cows on Mauna Kea, and they come from when Captain Vancouver gifted cows to Kamehameha back in the day, because he was one of our few like haole partners at the time. So this was us trying to go to the Waimau fence line. And then, and how Mikwa does it is after they fence off an area, they have to get rid of all the ungulates in that area. and then they start planting. So in the Waimau unit, we don't do planting yet because there's feral cows in the unit. Other thing that I experienced while I was up there is like I never drove on Mana Road for that much. So I remember this week we went one side of Mana Road, the Mauna Kea Access Road side, and came out on Waimea. And then this day we came from Waimea side, and it was just a cool experience being born and raised here and never going there before. It was a lot of like gallivanting all around the Mauna this summer. And some takeaways I had over the summer. On the research side, now MICWA they have some sort of research to base their decisions on for shade, shelter usage, and dibble size. Because when I started this project, I was kind of thinking like I'd be basing it off of someone's past research, but I couldn't do that, so I was kind of the first one to do it with Mauna Kea Watershed. But on a personal level, I kind of learned about like conservation work isn't really about like today, because you don't really see the effects of like planting your koa or like clearing out the ungulates until like a few years after. And so I included this picture. We built an extension to the greenhouse, and we're gonna put like gutters on it to collect water into a catchment tank so that a helicopter can come in and like get water for wildfire prevention. Oh, and then I decided to include this because it's kind of like representing conservation, how this building is gonna be there long after I'm with them, and it's gonna hopefully be there for as long as they need it to be. And I feel like that's exactly how conservation work is. And mahalo to University of Hawaii at Hilo and PIPES, the PIPES staff and my fellow PIPES interns. Thank you to the Kipuka Native Hawaiian Student Center. And big thanks to Mauna Kea Watershed Alliance for like Cheyenne and the field team for welcoming me in and teaching me everything you guys know. Mahalo.
Questions for Kason? All the way on the other side. Ooh, it's coming from your mentor. You better be ready.
Kason, mahalo for the presentation. It was great to work with you this summer. So the background of this project was looking at, we're seeing a lot of different weather changes in the past few years. So it's much wetter, like summers. And there's not a clear break in winter and between the wet season and the dry season, like it was maybe 15 years ago. So I guess my question is, based off of the data that you have so far, do you have a recommendation of what kind of shade shelters were effective, like as far as survival?
I found that the shade shelter was better than the yellow plastic one.
So historically, we got that from the refuge. That design was from the refuge. So it's good to know that it still works. The other thing we're gonna do is we're actually gonna do the same. We're gonna do the same survey at about a year, because we're trying to see if it makes sense to plant more in the winter. because we've been planting, most of the planting happens in spring traditionally, so with the changes in the climate. So thank you, Mahalo.
Ooh, one more question from another intern. I also am curious, since you're a Hilo student, if maybe there'll be continuity with the project and you getting to maybe outplant with them again.
Sup, Kason, Killer Project. I was wondering if the yellow shade thing that you did restricted the growth of the koa in any way? Like, was it preventing it from really like stretching out its limbs compared to the black shade cloth?
Based on like when I was out on the field, like even though they were getting restricted, they're still kind of growing well. But I did notice that with the shade cloth, shade shelter, since it's kind of just like two posts in the ground, they're more likely to like fall over in the wind. While these ones are more sturdy.
Okay, mahalo.
Can we get one last round of applause for Brother Kason? Up next, we have Miss Leilani Hoff, who's going to tell us all about our manu of the forest and how some of our cultural practices are tied to them.
Aloha mai kako. Hi everyone, I'm Leilani, and I'm going to be talking today about Kahili Pa'alima, some places that I call home are Wichita Lands in Texas, Muskogee Aina in Georgia, and Holuloa on the west side of Mokoke'awe. This summer, I've had the privilege of working with Dr. Kamelamela in the Hawaii Ethnoecology Hapua'a Living Lab, or the Hilo Lab. And my project is all about Kahili Pa'alima, which I will discuss what exactly that is momentarily. But I've been trying to understand how featherwork and Kahili Pa'alima are representation of Kanako Iwi relationship to place, ali'i, and manu, both historically and modernly. And right here I have a picture of Princess Nahi Ana and one of her Kahili Pa'alima. So what exactly is a Kahili Pa'alima? A Kahili Pa'alima is a smaller handheld kahili, most often used by female ali'i and their courts. Similar to a kahili ku, which is the large standing kahili you see at big state functions and in the Kahili Roman Bishop, they're a symbol of ali'i and crafted for ali'i by kahu kahili and held by a pa'a kahili. Kahili Pa'alima differ from kahili ku not only in size, but also as they were often carried by the ali'i themselves and not by a pa'a kahili. A Kahili Pa'alima in the hands of an ali'i functions as a scepter, as you would see in the UK or other Western kingdoms. Featherwork and the adornment of feathers was reserved for ali'i, as manu are closest to a kua, similar to our ali'i. Featherwork and feather adornments showcased an ali'i status. So here I have an image of Princess Ruth and two of her Kahili Pa'alimas. And then a little bit more background about my lab. So like I mentioned, I'm working in the Hale Lab with Dr. Kamelamela, and our mission is to advance knowledge that honors indigenous and local stewardship and that strengthens food security, social fabrics of society, community economics, and sustains biocultural diversity across Hawaii, the Pacific, and beyond. And why this project is so important
is there's never been any formal research done on Kahili pa'alima. There has been research done on Kahili ku. We know different bird species and materials used for those in Bishop, but there's very little published and accessible information about Kahili pa'alima. And our goal this summer was to change that. We did a lot of digital archives, a lot of working with Bishop, to create a database and find sources to share the mo'olelo of these Kahili pa'alima that are in Bishop's collections and beyond. So now a little bit more about me. Holuloa, Uptown 25 forever. If you guys didn't already know, I live on Duarte Road. If you know what Duarte, apologies. Holuloa is my foundation and my home. I spend every summer and New Year's here with my ohana, and then I have a video of me working on my Kahili pa'alima, which is here with me today. Holuloa is also one of my work sites. I practice feather work outside of Papahulu. I have Papahulu every Sunday from 12 to 4, and I do a lot of my research at home via digital archives and databases. So why Kahili, why feather work, why pipes? I have a lot of pictures of my kupuna in this section. I've been having more deep conversations with my mom as I've gotten older, and I found out this past trip whenever my mom was home with me last week that some of my kupuna did feather work, and I had no idea. It was actually kind of a conversation I wanted to have with my mom after Kumu Manai was telling us that you can always tell what physical traits you inherit, but it's more complicated to figure out what traits you inherited, like crafts and things like that. So this is the first photo as of my great-great-grandma, Alice Wentworth Kaleohano, and this is my great-grandma, Ka'awana Kaleohano, and this is my grandma, Leilani Young, my namesake. So 3 generations, and now I'm gonna be the 5th because it's gifted generation. But we're all doing feather work, and this internship has given me an opportunity to connect with my kupuna and learn something that I didn't even know, a gift I didn't even know that I had. So I'm so grateful for Pipes. Another reason why is I also chose to do this internship and chose to research feather work for myself, for my family, and for my mom, who's up there. And I also have a picture of my cousin Ka'awai Keakua, who I will talk about a little bit more in this presentation. So now getting into my ha'alele section. My halau is located right here in Hilo. When I started Pipes, my mentor gave me the opportunity to join her halau, which is Nahulu No'eau o Kanilehua, and my kumu is Kumugi Covington, and my first big project was this Kahili pa'alima. Having this opportunity to actually do feather work has given me the opportunity to actually practice what I'm researching and build a deeper connection with my research. I've gotten to learn Kahili inside and out. When it came time to name my Kahili, I was doing research about different names of Kahili. Sometimes the name is a description of them, or it's named after an ali'i or someone. So I decided to name my Kahili after my cousin who passed, Ka'awai Keakua. It just felt very fitting. So how have I been doing all these things and doing this research? In the first weeks of my project, I focused on building an understanding and relationship to Kahili pa'alima, not only through actually doing the practice itself, but I was doing all kinds of close readings from whatever Dr. Kamela Melo was sending me, as well as just like kind of finding things on my own through Papa Kilo and Ka'iwi Kilomoku. And I spent 30 hours on my first Kahili. And one of the sources that I came across kind of details the different parts. So I'll go over it and then I'll put the picture up here so everyone can also see. So the top part of a kahili is called the papale. The feathered portion is called the hulumanu. This bottom part, which is where my ribbon is called the pa'u, or the skirt. And the pole or the dowel is called the kumu. I also want to mention that kahili a lot of times, whenever they were being transported, would be taken apart. So the feathers could be preserved and maintained. But if the pole always kept the same name. So it's the pole that's given the name, and the pole will have the same name even if the feathers are changed. So if I were to change these feathers to black or white, it would still be Ka'awaya Keakua because the pole is the same. Okay. Now we're gonna go on a little huaka'i. So here is a photo that, I wonder where the map is. I have a map next to this, but this is a photo of Kuholalele. Two really important places in my research this past summer have been the Hamakua Coast and Ola'a. I took this picture in Kuholalele whenever we went on our huaka'i to Hu'imau. But the Hamakua Coast is famous and a well-documented place in history and in records for capturing seabirds. We'll get into which one was very famous. And Ola'a is known and famous for its forest birds and was known by kia manu and feather workers alike as a famous bird catching place. So now getting into some of the manu of these places. So Hamakua is known for the koa'ekea, which is primarily used in kahili ku. Those are the koa'e with the white tail feathers. And Ola'a was known historically as a bird catching place, specifically known for the following species used in kahili pa'alima and other forms of featherwork. And we see the i'iwi, apapane, elipaio, and amakihi here. And now I'm going to share with you guys some manu that we use now in contemporary featherwork. So in my work, I've only been using dyed goose feathers, as they are kind of like the most common that you will see being used in modern featherwork. You will also see hackle, which is rooster, the neck feathers on the rooster, pheasant, and peacock in featherwork modernly. Now going over some other past and present materials. So some common traditional dao / kumu / pole materials, kau'ila wood, koa, turtle shell, and human bone. Just for a little background, human bone is normally an ali'i's bone, and that's actually a good thing if you're an ali'i. The last thing you want is for your bone to be cut apart to be used for things such as like fish hooks and stuff like that. If your bone or your leg bone or arm bone is used for a kahili, it stays intact, and so does your mana. Modern dao materials are pine, birch, and commercially available wood. I have this photo here of some different kumus. So this one right here is painted conifer, going in order. The second one is ivory and turtle shell. This one is koa wood. This one is just labeled as laminated native woods. And this one is an old kau'ila spear. Now some more materials. So traditional cordage was olona, and modern cordage we use cotton or polyester thread. So here is an image of olona. Now kind of beginning to reflect and wrap up, getting into my ho'ina. These are just a few photos of my featherwork process and the ohana that I've gained from my halau. These pictures with all the black feathers are my first lei, my leivili poi poi, that I just finished on Tuesday. I'm so grateful for my mentor for letting me join her halau, and the halau has just been so kind and so loving towards me. I'm so grateful. And then I have two pictures right here. This one and this one are both from my first papahulu. This is my kumu Gay Covington. And kind of as my time with Pipes is wrapping up this summer, I just feel a huge sense of gratitude. I never thought that I would have the opportunity to do lehulu. And as a feather worker and as a researcher of kahili, I've created a new ohana within my halau, but I've also found a way to connect with my kupuna, and I'm very grateful for that. Now, I just wanted to say I'm super grateful for all of the new friends that I have gotten from this cohort. You guys are all so wonderful. I'm missing Uncle Koa today, but that was whenever we went to Seafood Boil. And I'm just super grateful for all of the memories that I've gotten to make this summer, and I'm grateful for everyone who's listened to me nerd out about kahili at any point or about featherwork at any point. I think the biggest thing that I've taken away is I know now that featherwork is a forever thing for me. It's a new kuleana that I've acquired. I want to continue doing featherwork,
and continue learning about Manu and Hulu in any and every capacity possible. Outside of my feather work, for my research side of my project, I have done many close readings about Kahili Pa'alima. I helped build a database with my mentor on Kahili, which will be pretty soon transferring sources and Kahili from Bishop into there. And I've created an annotated bibliography with over 25 sources. And I'm now working on a final paper that I'll be submitting to my mentor, kind of summarizing my findings this past summer. And then I just wanted to end, these are all of my projects. So the first one was Feather Pua. The second one is my Kahili Pa'alima Ka'oa Kea Kua. And this last one is my Levili Poi Poi. This was her just like an hour after she was all pow. And I actually decided last night, one of my great aunties, her favorite color was black, so I'm naming this lei Barbara for her. And I think another big takeaway for me was kind of expanding how we define Aloha Aina. I always kind of thought that Aloha Aina was just boots on the ground, hands dirty with soil, but I've realized that this summer your hands can get dirty within research, digging through archives, trying to find information, and from dyed feathers. My desk is actually stained orange right now because I'm about to start my kamoi, which is the flat, where the feathers lay flat. And I'm doing orange for that because that's my favorite color. But we aloha aina through our research by learning native birds and contemporary birds using Kahili and knowing their places. Feathers have been what has fed me this summer, both in spirit and by nurturing me through my research and my work. Mahalo nui, mahalo piha to all these programs and institutions who've made this possible, Hilo, PIPES, ASU, my mentor, Dr. Katie Kamelamela, my kumu Gay Covington, my halau, Nahulu Noeau Okani Lehua, mahalo. And I also have a surprise. Kiana, Lenalu, Vai, and Hinano, I have feather puas for you guys. So mahalo piha to you guys for everything you do for this whole cohort. Can we give them a round of applause, guys? I just am so grateful for all the opportunities that have been given to me through PIPES and for this ability to reconnect with Hawai'i and reconnect with my kupuna. But mahalo piha, that's all I have.
Amazing work, Leilani. I think you're just one of the many examples in this cohort of kupuna aligning things. You and Joshua, and there's so many others. We take so much effort into trying to make the best pairings, but this is none of our doing. It's all kupuna. So mahalo for highlighting them and bringing them into this place. And I'm just so grateful that you were able to connect on such a deep level in that way with them. But with that, do we have any ninau, any questions for Leilani? Another mentor question. Get ready.
Mahalo, Leilani, for all of your work. Being Hawaiian or growing up Kanaka Maoli away, I know was kind of how we started this. I just came back from Niagara Falls, Walpole Nation, singing this song to other nations, so I figured it's appropriate to do it to you.
Mahalo Piha for that. I don't have any words. Mahalo.
I think we could all use a little bit of a break after that. Can we get one more round of applause for Leilani before we take our break? It is currently 9:56. We'll be back in about 10 minutes with our next presentation. Quick bio break for everyone. up next, we have Mr. Kaila'au Pi'ena, who got to work with Hikiola this summer, and he's going to tell us all about the work that's been happening up Kealakekua. Oh, plenty guys missing. Where's your cohort? Okay, kaliiki, like ho'akahi minuke, alaila hiki ke ho'omaka. The floor is yours, Kaila'au.
Mai ka pi'ina kala I kumakahi, a I kona hawea hope ana ma o aku I ka molu olu o lehua. Ia ia e hulali, hele a ela I ka ili kai, a hiki aku I na lihi o na ina mau. Aloha nui kākou. I keia la, e ka ana like ka'u pāhana, no na ho'ailone I ke pili ouka. No laila, o wau no o Kaila'au Pi'ena, he kupa no ka mokopuni no Oahu, o Oahu. Ma ka moku o kona I ka ahupa'a o ka pālama, o wau ka muli o ka uahana, a me ka punahele a ka umaumakua o Iwalani laua o Wainwright Pi'ena. E hā o mākou, hele kula wau I ke kula nui o Bay o Ta'u ma Provo Uta. Aia hovi ko'u makahiki e kolu, or mana olana ko'u makahiki hope ma leila. Ke hele kula nei wau no ka huli kanaka, ka anthropology no ho'i. I keia kauwela, ua hana pū wau me ka hui o hana PIPES. Me Hikiola. Ua ho'omaka ia I ka makahiki, e lua kaukani ʻumikumaʻiwa. He uapo o Hikiola no ke kaiaulu e komo I ka hana ho'oulu la'au. A ulana I moana pāwehe o ka ike no na po'e āpau I pili na kumula'au ma Kealakekua Mountain Reserve. Malama lākou I umi kaukani mau eka ma Kealakekua. Kanu lākou I nā kumula'au like ole, e like me ka iliahi, ka naio, me ka ohi'a, e me nā ano mea kanu like ole. Lawe pū lākou I nā po'e wellness, mai doTERRA I keia wahi, e ho'omaka ika'i a o I ka mo'omeheu Hawai'i a me hiki ala kou ke ike I ka aina. Ma leila. No leila, o ko'u pahu hopu no keia kauwela, o no ka ho'oka'oi ka'u kahua, ma ka iki kupuna, a me ka launa kai ana me nā po'e like ole ma ka hana aloha aina. O ko'u pāhana, ua kupu ia ma ka pule mua, ua wala'au me ke kahi o nā 5 hana ma'o, ua mea mai oia kekahi o nā limahana ale ia I ike o ia I ke ano ka pihea I hoʻohana nā kupuna I keia mau mea kanu a lākou I mālama ai ma ka hale haʻakipu ai ole nā moʻolelo e pili ana I keia mau mea kanu no laila ua kupu maila kekahi noʻonoʻo I nā hiki ke hoʻokumu I kekahi hoʻailona no nā mea kanu a pau a lākou e ulu ai ma ka hale haʻakipu I ike leʻa ma ka ʻaina o ke alakikua mauka I hiki ke lole I kekahi kahua paʻa punai wele me na ike like ole no nā poʻe kamaʻaina a me nā poʻe malihini no laila ua pono wau e haʻalele mai uka ike ke ena luʻu I loko ka nā puke a me nā moʻolelo like ole no laila ua ohi wau I nā ike pili mai nā ano wahi like ole ma ulukau ma wehewehe ma Bishop Museum Ethnobotany Database o kela nā kahua o ka'u mau ike a laila ua helu pū I ke kumu au puni loa kekahi mau moʻolelo hoi a I nā kahua punai wele e aʻe ua kōkua pū nā limahana ma KMR iaʻu e hiki ke kamaʻaina I nā mea kanu a me nā kumulaʻau like ole no ka mea ano koʻa ko lākou hiʻohiʻona mai nā mea kanu a me nā kumulaʻau au iki ai ma Oahu no laila he ua paʻakiki no mau no koʻu auana I kēlā o kekahi moʻolelo hoi aʻu iaʻo ai au I helu ai ma ke kumu au puni e pili ana I ka sandalwood trade ua kaʻana like I nā poʻe Hawaiʻi I ka iliahi o kapa ia ma ka puke ka laʻau ala ua kaʻana like lākou me nā poʻe maikaʻina e no nā mea lole no nā mea kaua ua kauoha ia nā mōʻī o Paiʻea laua o liho iā lākou nā aliʻi a me nā makaʻainana e piʻi uka ohi oki nā laʻau ohi ia I hiki ke lawe I nā poʻe maikaʻina e ma mua ko lākou halihali nui I keia mau laʻau ma luna o ko lākou kua ua kupu maila kekahi leho ma ko lākou kua no laila ua kapa ia ke mau poʻe nā kua leho so he mea hoi no ia o kia koʻu kaʻina no ka hoʻomākaukau ana no laila ua hoʻolālā I kekahi olokeʻa no nā hoʻailona a me nā ike pili I iminoʻi ia ua iminoʻi wau I pili ana nā moʻolelo nā ʻolelo no ʻeau pehea I hoʻohana nā kupuna I keia mau mea kanu imi kekahi a laila ua imi kekahi wahi no ka hale e kau ia I keia hale no nā hoʻailona a ua wale ia o keia nā ano I ke pili I ohi ai so laila nā inoa ko lākou wehewehena ko lākou nohona nā mea I hoʻoweliweli ai I nā mea kanu ko lākou mau pakanā pehea I hoʻohana nā kupuna ia mau mea kanu I nā hoʻohana no ka laʻau lapaʻau ka lei no nā aha he hana moʻolelo nā ʻolelo no ʻeau nā oli a me nā mea lei o kia nā mau mea kanu aʻu I iminoʻi ai aia ka iliahi, naio, mamane, pilo, pukiawe, ala wainui, paʻiniu, pela wale aku. Au keia kekahi kiʻi o ka hale haʻakipu ma laila. No I koʻu iminoʻi ana o keia koʻu manawa mua I ike leʻa I keia mau inoa, keia mau mea kanu, ale wau ike ai ma mua koʻu hele ana ma laila so ua nui koʻu e aʻo ai, kau e aʻo ai. Ua huʻakaʻi mau ma luna o kēlā ʻaina, he nui, a ua leʻaleʻa no. O keia kekahi kiʻi o mākou e piʻi ana I uka e ohi ka laʻau naio. No laila, o kekahi o kaʻu hana ma laila, ua ohi ka laʻau naio I hiki ke lawe I ka hale puhi no ka puhi ia no ke kahi aila, no ka mea ua kuʻai aku ka aila ia doTERRA. Ua kōkua ia kumukele i ka noi me kona mau papa hoʻonui ike ina hiki ia ʻoike ike ma laila. Ua hapai mākou i nā mau upena i hiki ke hopu i nā manu o iwi. O hoʻaʻo, but ʻaʻole i holomua. But ua kipa maila o Brett Mossman, kekahi hana, kekahi poʻe hana ma ka mena mea, mena manu o iwi. Ua hana i nā mea like ʻole mai ka hoʻomakaukau i nā moena. Ka hoʻomakaukau i nā pāmeaʻai no nā poʻe e kipa mai ana. So, ua pono wau e hoʻolohe i koʻu kumu a hana i nā mea a pau. A kekahi mea koʻikoʻi au i kukulu i kēia kauwela o ino ka pilina me ka ʻaina. O hiki aʻu ke ike leʻa i nā manu o iwi. No i koʻu manawa i noho ai ma Oahu, ʻaʻole au imi no nā manu o iwi. No koʻu manawa mua ka ike ana i kekahi ʻio, e lewa ana ma ka lani lā, ka lohe ana i ka leo, ke kani o ka manu ʻamakihi a me ka ʻapapane. Noho i he mea koʻikoʻi no ka mea ina hiki aʻu ke ike leʻa mau popo i ko lākou olo ana. Ua kukulu i kekahi pilina i hiki ke paia no lākou no ko lākou ola ana. Iaʻu he mea koʻikoʻi no nā poʻe a pau e kukulu i kēlā pilina i ike leʻa iā lākou. Ua hiki ke kukulu i kekahi pilina me ka ʻaina o kona ma ke ala ke kua. He mau moʻolelo, kaulana no ia wahi. Ua loa nā kahe mau me ke kōkō nā aliʻi kaulana ma o. So, haʻaheo no hiki ke noho ma laila i kēia kauwela. Kekahi mea hoihoi aʻu iā ʻo ai pū, kekahi fun fact no hiki ola. Kanu lākou i kēia makahiki i 8 haneli, 18 mau kumulaʻau mai hiki ola mai no nā hoa kipa. No ke ala ke kua Mountain Reserve, i kēia makahiki wale no ua kanu ia i 10 kaukani, 13 kaukani e hiko haneli, kana hiku kuma lua, a i ka pau ana ka makahiki e kanu ia ana hoʻokahi haneli, 10 kaukani mau mea kanu. Mai ka makahiki 18 o 18 ua kanu lākou i e hiku haneli 11 kaukani e ono haneli kana 19. Pai lima no lākou. No laila, o kēia ka'u i ohi ai kekahi la'ana o nā mea ike pili i ohi ai mai no ke koa, he mau mea like ole. A laila, o kēia ka wahi i waele ia no ke kukulu ana kekahi hale, ʻaʻole hiki ia mākou ke kukulu i koʻu manawa i kēia kauwela ua paʻaia me nā wahi mea like ole. A kāna naʻe o kēia ko mākou mamake no kēlā wahi. Kukulu loa ua kuʻai e lākou kekahi kauhale o kekahi hale. So pono mākou e just hana wale no. Ia no naʻe haʻaheo wau i ka hoʻomaka ana i kekahi i ke kahua no kēia hana. O kēia kekahi laʻana o nā hoʻailona ui hana. Loa na inoa, ka inoa Hawaiʻi, ko lākou inoa epekema, ko lākou inoa laha, a me nā inoa e aʻe kekahi ike pili mo o meheu, me kekahi ike pili kaiaola. Mamake kau i kekahi QR code, i kekahi wā e, i ke hui i ka punai vele e Kemar. I ka hila ana ka naio me ka ie, a me ke iana mea kanu a pau e ae. No laila, o ke kahua maumua, ma hope ke kukulu, i ko'u manawa ma'anei e keia kauwela pōkole, ua i ko'u manao, o ano mina ma mua a kano nae, ua haʻeheo no i ka ke kukulu ana ki kahi kahua no keia hui, hiki a lākou a mena poʻe aʻe, ma hope a lākou, hiki a lākou ke kukulu i ka hale, o ka ike a me ka nao au, no nā poʻe e kipa ana, ia wahi. Mana o lana hiki au ke hoʻi hou, maila i ke ia wahi ma ke alakikua. Komo pū i ka hana, ko lākou hana ma o. Aia no i ke kō a ke au. Nā ka manawa no e a'o mai ia'u i ka'u alahele. Kau ko'u hilina'i ma kia kua e alaka'i mai ia'u i ka alahele kupono hiki a'u ke ho'i mai i ka mole. A kokua i nā po'e mai ke kaiulu Hawai'i, ana po'e a pau. He i'ini ko'u e ho'o lawe i ka ʻaina ma'ane'i. No laila, he mau, i keia kauwela, ua hiki ke ike i nā ano i nā puka like ole hiki a'u ke hele ai. Ala wau i koho kia manawa a ka maupopo no wau e ki kahi wā e hiki mai ana pona wau i koho a e maupopo i ke la wā, i kia wā, o komo wau i ka hana a ohi ai ka ike a hiki. Mamake wau e mahalo i nā poʻe a pau i nā limahana ma Kemar, ma ke ki'i ai no o Brian, no kona ai, like, keu a ka ono ma kona. Me Greg, a me Kumukeli'i Kanoi, ma'ane'i, me Kamakani, Willie, Poita, Jojo, Amanda, Hoku, Kioki a me Keoni, a me Shellen. Mamake mahalo pū i nā wahili mahana o Pipes, o Shannon, Kiana, ko hana, ko vai, me kona ohana, o Sharon, kala mai. A me Leinalu, a me Hinano. Mamake mahalo pū i ko'u ohana, ma oahu nei, e ka ko'u mai ia'u, a me ka ohana Sata Ellis, no ka lākou malama mai ia'u ma keia mokupuni nei. A me nā hoaloha i ka ko'o mai ia'u i lawe a'u ma o aia ne'i o ka moku nei, me ka ole o ke ka'a. Mahalo nui, a me nā wahi hoa mai ka ohana Pipes. Mahalo nui o ko pākahi a pau, o mākou nā alaka'i hou o keia au. E alaka'i ka lāhui Hawai'i i mua. Mamake pū e mahalo i nā sponsors a pau. Malalo nei. Mahalo. No ni no. Ale pono, but.
Aia ke kahi ni nao mai a Shaylee mai.
Mahalo nui. I ke hoʻike ike. He nino ka uno ke kahi o nā ki'i I hoʻike ia ma ka hapa mua. He ma ka mua loa paha. O nā kane ma like koko ki kela. He kahaki pa kela ai ole he ki'i
maui. O keia. Oh yes.
He mo'olelo paha
he
kona?
He ki'i no ia. Aia ua kahaki ia e Ed Caton. Aia no ma ke ena ko mako wahi. Aia ke kahi mo'olelo ma luna o ke la ki'i. I think ua aia o mau peailu. Yeah. Ua hele lākou I uka. I think ke kahi kona o ike oia I ka aha no ke oki ana I ke kumulao no ka laiwa'a. No leila ua I ke kahi ki o ko lākou hana. Ale mau popo ma ka mahea. Ma honau nau. Ua hana ke ia so.
He wahini ele wale no. Mahalo nui.
Mahalo kumu.
He mau ninau e a e no kaila au. He ninau ka'u. Ninau ano pa'akiki paha. He aha ka vai o ka ho'ola ano ka olalo ma kia ano po'e a pili.
So hiki ke ninau.
He aha ka vai no ke hala ho'i oi hana I keia hana ma ka olalo Hawai'i, ka olalo makuahine, ka olalo keia aina. He aha ka vai ma laila.
Ua vai no ka mea keia ka olalo makuahine a ui hanai ia ma ka wā papamalao a hiki ka upu ka kulana ma ka meha. But I kou manao he wahi o lelo like I ha'i ia uiho I manawa a pau you know ina ho'o ke kahi mea pona I hana mau I ma'a mau I keia hana you know he mea veli or this pa'akiki ka ololo Hawai'i ke kahi manawa ina aole ho'o ma'a e mea hila ke kahi manawa a kano na'e e holomua wale no e hoa'o a hiki ka la e puka or just pi'a e ko makaukau he mea vai no na po'e a pau e a'o I ka lolo Hawai'i no ka mea ina hiki a oi ke a'o ina olelo hiki a oi ke apo ina ano kaona like ole ma na inoa o na manu a me na mea kanu a me na mo'olelo o konu pepa a me na mea a pau no leila. Yes, mahalo.
Mahalo, Kaila'au. Can we get one more round of applause for him, please?
Hello. Aloha kako. Let me get situated here. Mahalo to all of you here in person and online for joining us for these presentations. Our next speaker is Koa Collins. He is, however, battling some health issues right now and unfortunately unable to join us. And instead of moving along because the work that he did and where he did it and with who he did it was highly impactful in a myriad of ways, we'd like to share about him and about his project. And in the near future, we will have an opportunity for him to present his story map, and we will inform all of you of the details of when they're finalized on our media platforms and via email. So I asked his mentors and him to send me some photos that we could share and kind of click through. If it will let me, yeah. As I talk about Koa, I'll also share some things that he sent me that he'd like me to read, as well as a lot of messages of aloha and reflections from his host organization and his mentors. Okay, so Koa is born and raised in Kona. He's a Kona boy through and through. He's finishing his degree in liberal arts at the Hawaii Community College Palamanui campus. He's a student, a surf instructor, and a kupa of Kona, with a growing pilina to the kai and aina there, which I'm convinced has deepened exponentially over the last 10 weeks. I didn't write this part down, but he's maybe my favorite intern story this summer. I did one class talk to one class at Palamanui campus, and he was in that class with Kumudru Cap. And and that's why he applied to PIPES. And he's a non-traditional student studying liberal arts. Stepping into an aloha aina conservation space was very new to him, but he was so excited and so enthusiastic and so curious. So I think that's just setting the space for me to share about from his mentors some words of aloha and reflections about him. He was working at Ho'ala Kealakekua Nui this summer in Kealakekua Kona, where they are actively restoring stewardship practices and restoring actual sites of the Kealakekua coastline, Hikiau Heau, as well as doing reef monitoring and fish surveys in the kai. They also recently acquired some aina up uka that they are starting to steward as well. And because of the big surf during the summertime, and it was pounding in Kona this summer, Koa did spend a lot of his time up on the uka property, just manhandling the California grass and being the hammer that we all know he is. So I'm gonna just go into these from Keanu, from their team. They say, spending the summer with Koa has been nothing short of inspiring. He is one of the most solid kanaka I've had the privilege of working alongside in a long time. And throughout the summer, he taught all of us just as much as we hoped to teach him. One thing that always stood out to me was his willingness to step into every opportunity without hesitation, no matter the task. Koa never hesitated the work. His curiosity and determination in learning guided his actions, and because of that, I have no doubt he'll overcome any obstacle he sets his heart on. His passion for aina, community, and ike kupuna is in everything he does. He carried himself with humility, worked hard, asked thoughtful questions, and consistently showed what it means to grow into the role of a hoa aina. Koa has a heart of gold and even stronger drive to become the best version of himself. I'm incredibly grateful to have had the opportunity to work alongside him this summer and can't wait to see where his journey takes him. I know he'll continue to make a meaningful impact wherever he goes, and we hope to see him back at HKN in the near future. And to Kanani Inos' words, Koa has been an amazing addition to the HKN Ohana, seamlessly slipped right into our daily hana and routines. His willingness to learn and participate in all of our activities and projects made him a joy to be around and a real gift to our organization this summer. I admired his calm, aloha-filled nature and his dedication to the aina and kai, always fully present and ready to kokua. I enjoyed his meaningful contributions at our staff meetings and opening blessing of Hale Kūpehu, our Ohana Koa Day, and other work events and activities at the Bay and up at Hale Kūpehu. My last interaction with him doing some clearing work at Hale Kūpehu, where he mentioned how he truly felt a part of the HKN Ohana and our Vahipana and saw himself there forever. I deeply felt and appreciated his work, commitment, and aloha, and sending prayers of gratitude for him coming our way this summer and pule of healing and comfort at this difficult time. Our deepest aloha to him. Kahaka'io's words, Brada Koa from day one has been the definition of one hammer. Not only willing to huli na lima ilalo and get down to work, he also brought in his past experiences, knowledge, and mana'o to take our ohana above and beyond. Even as an intern himself, he was a role model and alaka'i for our younger high school interns. Brada Koa exemplifies aloha aina, mauka to makai. I'll just say here, HKN had a couple of Konawaina interns, so high school boys that were down at the Bay with them, and Koa really took to being like an older brother to them. So it's cool that as an intern, he got to kind of mentor other interns. And kind of his primary mentor, Kainalu Keli Kuli Grace, who is a PIPES alum as well. These are his words. The first field day of Koa's internship was during one of the large south swells, and the bay was pumping. Me and Koa were doing some shoreline kilo, and I asked, "What, you get your board?" He replied, "Let's get out there." We sat in the lineup at Hale pole break and talked story and traded waves for a few hours. As the tide began to drop and waves started slowing down, a large Eva circled about 20 feet above our heads, making loud noise. We both looked at each other and instantly took it as a ho'ailona of warning. We caught the next wave in as fast as possible. And as we were getting settled on the shore, noticed the Eva headed toward the pali where I believe its nest resides. Looking at it now, I don't believe the Eva was a warning for us, but a ho'ailona of ho'iho'i for Koa, because like the Eva, after a long journey, Koa has returned home where he belongs. The internship I had planned for Koa and the internship he experienced were two very different things, yet he showed up every day with a smile on his face, working hard, learning, and teaching. Mahalo piha, Brother Koa, for being so solid, and welcome to the ohana. We look forward to many more amazing experiences with you. And then from Shane Okoni Nelson, Uncle Okoni as we know him, Koa joined Ho'ala Kealake Kuanui's Hoa Aina Ohana, engaging with passion that never ceased. His love for Aina, Mo'omeheu, and Kaiaulu is evident in his work ethics, desire to learn, and his natural ability to become awakened to the surroundings and contribute to a collective kuleana. These characteristics are acquired that build great leaders in our community. Koa is an exemplary learner, and during his time with HCAN, a mentor to challenged youth who are struggling to find their place in community. Koa's drive to dive deep into how HCAN practices community-based conservation management that is led by place-based traditional ecological knowledge has broadened his scope on how he sees the world and brought depth to his already existing values, skills, and personal philosophies. It is a ho'aina like Koa that our partnership with PIPES is meaningful. Oops, that was the last one. Just hanaho. Koa and I had discussions about his presentation, and I spent time with him in the field. I believe his goal for the summer was fulfilled, as so did HCAN's goal with Koa. Therefore, I congratulate him on a successful internship with HCAN. We hope to continue our work with Koa in the future and wish him the best of health, prosperity, and success on his personal journey. The last thing I'll share is what Koa wrote himself and would like to share with all of you. I'd like to thank everyone from PIPES staff, my fellow interns, my host organization, HCAN, and all the other organizations I've had the privilege to work with this summer. It's thanks to everyone's support that I've grown a deeper connection to this community and Aina. Unfortunately, I'm unable to be there present this last week of programming. However, I'm forever thankful for the connections I made and the knowledge I gained. It has set me on a path to become the kind of kupuna I know my family and community can be proud of. Although I can't be there, I hope that others can share a bit of the experiences that we had together. Mahalo nui loa, Koa Christopher Roy Collins. Mahalo piha Koa. It's also his birthday today. Happy birthday, Koa. I know you're watching. We love you so much, and we're excited to see your presentation in the future. Mahalo. Back to you, Kiana.
You hit F11 while you're up there real quick. Thank you. Up next, we have a keiki ko'olau that has turned into our temporary keiki kohala for the summer. Miss Ko'iahi Punua, who's gonna take us to ko'uwahi aina puna hele, ko'u aina no ia, me lele o kohala me he lupe la. Excited to hear the stories and probably have a little bit of tears too. But take it away, Ko'iahi.
Aloha, everybody. This is my pahana, Lele o Kohala Mehelupela, mapping through Mo'olelo in Kohala, Hawai'i. O ko'olau poko ku'umoku, o kane o he ku'a hupua'a, o puna lu'u ku'u'ili, o ka āpua kia ku'ua. O ka ulumano ku'umakani, o kia hia ka hoi ku'umauna, o na one o la'a ku'aione, a o ka waho kamano ku'ukai. Aloha, everybody. My name is Ko'iahi Punua. I'm from Kane Ohe Oahu, and I wanted to share these places and these elements with you because, to me, this is who I am. All of these elements encompass who I am as a person, and I hope that I am a good reflection of my Aina. And I share these places with you because growing up, they weren't just names, but they were also mo'olelo that brought life to these spaces and shaped the way that I aloha aina and the way that I interact with the aina. And so I know that anywhere I go, sorry guys, I told Kiana them that I wasn't gonna cry, but anyways, I love my aina. It's my piko. And I know that anywhere I go, I know where home is. This also brings me why I wanted to join PIPES. I never heard of PIPES until I think it was Leinalu who told me about it, and a couple other people who I knew had done PIPES previous to this summer. And I kind of saw it as a way to learn from mentors, also from my fellow interns, and the amazing alaka'i that we have here at PIPES. And I also saw it as an opportunity to grow personally and professionally while be able to do hana in the places that have nurtured me and cared for me. And yeah, it's been a great summer so far. So I'm really excited to present this. My host organization was Huliawa Pa'a. My mentor is La Crivello. Huliawa Pa'a is a nonprofit organization whose mission is growing Hawaii's community through culturally based forms of innovative learning, leadership development, and collaborative networking in Wahikupuna stewardship. Their vision is Wahikupuna thriving through Hawaiian self-determination, and they partner and collaborate with various community groups, educational institutions, archaeology firms, and lots of kaiulu throughout Hawaii with the purpose of creating a safe space for Wahikupuna stewardship. My project focus this summer was to initiate the development of a geodatabase for the North Kohala community and to explore how these traditional knowledge systems, ikekupuna, can be translated through various forms such as mo'olelo, mele, oli, and to show just how these forms of knowledge function as maps and can be integrated through GIS with ikekupuna to demonstrate how they can complement and strengthen each other. By doing that, I'm going to be doing that through the mo'olelo, Hano, the birth story of Kamehameha Pai'ea. And it kind of just serves as the case study for my project to demonstrate how mo'olelo can be mapped and how the mo'olelo is the map. And to just acknowledge the understanding of mo'olelo as a map in their own right. For my ha'alele and my methodologies, a big part of this project was just going out, being with the kaiulu, being in the aina of this place. I had the really, really amazing opportunity to live in Niuli'i, Kohala, which is like inside Kohala, north Kohala, with my auntie. Shout out to Auntie Lehua, Uncle Ashton, and Iti Girl for letting me live in their hale with them this summer. And so just being in that aina itself, I knew that I would have to go out and do my own research and kind of get a better feel of this aina, especially if I was gonna be doing work here and living here. So these are just some pictures from this summer. This is my mentor, La. This is us. This is me at one of our first or like second field day. This is in Ka'u. And then this is just the Huliawa Pa'a Hui. This is a picture from Ako'ako'a Point in Kohala. And here you can see Avini, which is like where the pine tree is. And then just more field day works. Also, I had to look through old maps. So this is let me see if I can click on it. This is an old TMK of North Kohala. You can kind of see those Ahupua'a boundary lines and just this old map that I think is super cool. Yeah. And I also incorporated the Kali'o Kapa'akai Collective, their values and methodologies. This is an interdisciplinary community of practice, and my organization, Huliawa Pa'a, is the backbone organization for this collective. So I tried my best to kind of incorporate all of these values within my summer. Okay, so getting into kind of the hana hana and my huaka'i this summer, mahalo to my mentor in the kokua in creating these amazing maps. So this is just a map of Hawaii Mokupuni, and then we go into this map of North Kohala, and you can again see a lot of the ahupua'a boundaries. And in North Kohala, there are over 30 ahupua'a just in this part of Kohala itself, and a lot of them aren't mapped just because of how tiny these ahupua'a are, so they get smushed into like the biggest ahupua'a adjacent to it. So not all of them are mapped, which is another project that is gonna happen. But yeah, so it was really cool just learning how to do these maps. I still don't fully understand it, but I had to do trainings to get myself familiarized with the type of technology that we're gonna be using. And then again, kind of going back to my hua and getting to know Avahi, I think that the makani and the ua are a big aspect of a place and getting to know people of place. So here is a map of the makani and ua that I was able to find through Hanau Ua Winds and Rains, great puke that everybody should go read through to find makani and ua of your aina. But we were able to create this interactive map. So if you click on, the makani or the ua, it'll say if it's a wind or rain, kind of the description of the rain, where it's located, the direction, and then on this side is again with the descriptions and the rain. But there were so many. This is just the ua, and then these are the makani. Two makani that I wanted to note was A'a and Kaumi. These two makani I actually found in the Nupepa Ku'oko'a in a column called Nahi'ohiona o Kohala. And so they talked about how A'a and Kaumi, A'a is Kohala Loko and Kaumi is Kohala Vaina, but they're both kind of lighter winds of the Apa'apa'a, which is a well-known rain of Kohala. And then for the bigger portion of my project was doing research and mapping the mo'olelo hanau of Kamehameha, the birth story of Kamehameha Pai'ea, and Nai'ole's huaka'i throughout North Kohala to save a king. I wanted to look at multiple sources of information from this mo'olelo, and so one source was an oral history by Anakala Fred Kachola. This source, a lot of the information was previously gathered by my host organization, and so on the side over here you can kind of just look through if you want, and it'll take you to, whoa, wait, how do I go back? Okay, sorry. Okay, yeah, sorry. But you can click through these links, and there are also other sources of information to watch Uncle Fred and his mo'olelo. So my kuleana with this source was, again, reading through previous interviews, watching videos of him, and then being able to map and put descriptions to the places that he noted in this mo'olelo. So in his mo'olelo, it starts off in Kokoiki, and then we go to Ho'ea, which is Ho'ea Moku. It's actually in the ahupua of Kahei. And then we go to Hawi, Honomaka'u, Kapa'au, Halaula, Halava, Makapala, and then we get to Avini. And so we were able to just map out these ahupua'a and kind of make a makeshift path that Nai'ole could have possibly traveled. And then we were also able to get this map that Anakala Fred drew of the mo'olelo that he shared, kind of what he thinks the route was to get through from Kokoiki to Avini. And so I'm not sure if you can see, it's a little bit blurry, but yeah, I just really wanted to include this, especially since we are mapping this mo'olelo, but Uncle had already drawn one, so I really wanted to add that in here. Again, there are many sources of this mo'olelo that have been preserved through different ohana, different kaiaulu, and different sources of ike, and so I really just wanted to show and reflect these different perspectives and sources because they're all valuable and they're all important. Another source that I really wanted to include was Nupepa. I think that Nupepa serve as vital sources for perpetuating and preserving mo'olelo, iki kupuna, and just perspectives of our kanaka Hawai'i and our kupuna. You can learn so much from our Nupepa Olelo Hawai'i. Okay. So this Nupepa here, this is O'u I Kamaka o Kawauke Oi o Pio Pio, Kamaka Stillman. She is a kupuna of Kohala. Her great-great-grandmother is Kaha Opulani, who was the ali'i wahine who nursed and raised Kamehameha Pae'a in his younger years. But, oh my gosh, I found this Nupepa. Sorry guys, I'm gonna cry again, but. In this Nupepa, she says, na ue o laloa e, na ke kohala loko, kohala waho, ke kohala ua a pa'a no ka mea, ua nohoa a kupa a kama'aina a ewe a mole a kohala o ko uone hanauia. Sorry. Again, when I found this, I got very emotional because this is how I feel about my aina, and so, yeah. But when I read this, I just kind of took this as, this is a mo'olelo that I really did want to include because, especially in this specific newspaper, she's kind of going back and forth with the author, and the author has the story of Kamehameha's birth, but she's saying like, you're wrong. And then she, she's saying like, you're wrong because I'm from this place and I know this mo'olelo, and she also notes within this mo'olelo, her mo'oku auhau, Kamehameha's mo'oku auhau, and then she always signs off with something similar to this, but just saying how she is Kohala. She's not just from here, but she is a piece of this aina. And then this is a picture of her. Oh, can I click up? Yeah. When she was writing this specific newspaper, this message, she was 83 years old. This was written in 1911, and she passed away in 1924 when she was 101. So I think also just noting how sharp her memory was from the mo'olelo that she learned from her kupuna. But yeah. So hopefully you guys can go back and look through, because I think even just the olelo in this is amazing. She says all kinds of things that I'm like, I never would have thought of that. It's so cool. But yeah, I really wanted to include Nupepa, and then also in her source, she says that Kamehameha is born in Ainakea, which is again different from what Anakala Fred was saying from when Kamehameha was born in Kokoiki, but I kind of just highlighted specific things that I thought were important to note out of all of the amazing things that she said, but you can just look through them later. But it was really awesome reading through this mo'olelo and being able to read through both sides, the author's side and her side. Yeah, it'd be awesome to take a look. And then another thing that I wanted to add was also Mele and Oli, because I think they do really function as living archives, and they are oral maps. And being able to kind of look through this as a historic record that goes beyond the physical modern cartography. And so this Oli is Eia o Avini Paliali Ihula Ana. It was shared by Kumuhula Lorna Lim and her kako'o Lokelani Hutin. And so when Kumu Lorna shared it with us, she olied it for us, and I kind of remember seeing it somewhere, and so I went back into the Nupepa, and I found it in the obituary of Kamaka Stillman. and she like had it all written out over here. And so I just thought, again, what an amazing source. And I think the reason why I didn't necessarily map this specific oli, or even what Kamaka Stillman was saying, was because it's right here. This is noting exactly what happened, at least from her source and her experiences. It didn't say who wrote it. I'm assuming maybe she wrote it or her kupuna wrote it. But I really wanted to include this, especially because mele and oli really, you can learn about a place through mele and oli, and it's extremely important in our ike kupuna and kona ike Hawai'i. And then just coming back to ho'ina and ha'ina and just reflecting on this summer. It's been a really great summer, and I think especially learning from my mentor through things like GIS and modern technologies, and kind of finding a way to show that they are not replacing our ike kupuna. A big lesson was that technology should always remain secondary to pilina. I think that pilina aina, pilina kaiaulu, it's more important than, yeah, and when it comes to things like maps, they're only meaningful when they reflect the ike and leo of our kupuna and who we are, because if we didn't know where we were, there would be nothing to map. So, yeah. My ka'au doesn't end here, but it will continue through the work that I do and the work that we will do. I'm so sorry, guys. I didn't know I was going to cry this much, but yeah, this has been a really great opportunity to just be here and also be with my ohana in Hawaii Island. And so, yeah, mahalo nui. I just also want to thank, again, PIPES, all of my amazing friends and family that I've made here. Huliawa Pa'a, my mentor-in-law, and just the ohana that they've given me there. In the last week of my internship, I got to be with their Wahikupuna internship program interns. And so I learned so much just from that week. And I was so happy that they welcomed me. And I kind of got to double dip and be with PIPES and Wahikupuna internship. And then also, mahalo to my ohana. Yeah, thank you guys for everything that you guys do for me. And mahalo. Sorry for all this crying. I did not think I was going to get this emotional, but mahalo. Any questions?
As I make my way to that side of the room, I just want to mahalo nui ya'oi e ko'iahi no ke'ia hanana. And I'm not going to look at you when I say this, because I think you really embody what Kalani Kuali'oliho said when he said, "Nā vai ho'i ka ole ake akamai, he ala I ma'a no I ka hele ie umauma kua." And the work is just living proof of that.
You guys did a really great job. I've been a witness to this little one, and I'm so excited that you're gonna be better than we are now. And I'm over here like, kill girl, like work. Like, you just starting. And Ko'iahi, I'm really looking forward to working for you one day. So like, just remember, we're just here to support, and it's just been so amazing. I was like, what are these keiki ko'olau gonna do? Like, what's gonna happen? And it's literally just starting. And we had a special thing that happened out in Kohala a few years ago. Those of us that were there. How do those type of pilina fit into these type of mo'olelo that you're recording nowadays? We don't have to tell everybody what happened, but you was there. And you was at the ceremony. So where do those experiences and pilina fit within our mo'olelo GIS? I don't know if histography is a word, but I'm just gonna make it up. But so proud of you, so beautiful. Just keep shining. Okay.
Thank you. Yeah, I think definitely pilina to aina is incredibly
it's so important. And from what you're talking about, yes, I was there, and that was just such a magical experience, especially when you can hoʻihoʻi kupuna to their aina. I think that these mo'olelo are just snippets of those people's lives because they are the ones that share this mo'olelo. And so when it comes to mapping, when it comes to putting it through a mo'olelo, I think that the first source should always be the person of place, because they are what makes it valuable. So,
yeah. We have another Nino on this side.
Hi, Tilla.
Hey.
So we've spent most of the summer just talking about how mo'olelo are maps. And I think when we first started, I told you I'm a GIS specialist, but that's just because my job is pilina. I like mapping is pilina. GIS is just showing relationships. When we think about that, if pilina is the priority, how have, our kupuna are our maps, they're our map makers, so how have you taken on the kuleana of being a map maker? And what do you feel that that has done for you in this time over the summer?
What a great question. I think that now that I kind of have all of these experiences, I want to take it back to my kaiāulu and kind of implement what I've done here and do it within my own community, which is, again, a big part of what my mentor is going to do. This is just the start of a bigger project throughout this island, throughout our Pai'aina. And so, yeah, I think being a map maker, or learning how to be one, I'm going to try to just implement this within my own kaiāulu and hopefully be able to interact with more kupuna who are willing to share their mo'olelo so that we can do something similar to preserve their ike and all that they have to share.
Any last comments from Ohana, who came all the way?
No.
Ole paha. Well, can we have one more round of applause for Miss Ko'iahi? We're gonna stay makamoku e lele ai mehelupe la in Kohala with Miss J Boys next, taking us to the work that's happening up with TKC and the nice cloud forest.
All right then, aloha mai kako. How's everyone? I'm Jay Noelani Boyce, and today I want to talk to you guys about what I've been doing up the Kohala Mountain. So I titled my project Malama Kohala, since that is what I've been essentially doing this entire summer. I have been taking care of an aina that is taking care of us. So to start off with a little bit about me, I was born and raised here on the Big Island of Hawaii. I have family all over the island, from Milili'i all the way to up Kohala and Honoka. I am an incoming senior at Hilo. I am studying environmental science, and this is my first time being a PIPES intern. And I was put into the pathway of aina, which I'm really proud of. So a little bit about me. I'm very family-oriented. I love spending time with my family. They're a very important part of my life. I love being outdoors, hiking, fishing with my dad and brother, visiting my family up in Honoka with my grandpa, who is 104 this year. And also hanging out with friends from Waimea. And you kind of forget about the pets in your life. This is my dog, Rocket, and my lovely cat, Katniss. I've always been really outdoorsy. Always loved going outside to the ocean with my dad. I've always been the weird kid that grabs the biggest crab and chases everyone around while everyone's screaming. And just, appreciating what is given to me from the ocean. I also love going up to the mountain. I love going up Mana Road with my family, we have crazy stories from up on the road. I know this time we went up here, we had one coal tree that fell in the road, so we took like 3 chains and a 4-wheeler to move the thing out of the way because no one had one chainsaw. And I know this other one, my auntie was cooking rice while going up the mountain in a Ford Expedition. One Portuguese lady. So moving on to my host organization, I was partnered with the Kohala Center this year with my mentors Brooks and Jake, as well as the crew Ben, P.E., Jay, and Megan. And the Kohala Center has been such a wonderful place to work at. Their mission is to hanai pilina, to respectfully engage the island of Hawaii. Their vision is a state of pono as revealed by Aina. They were founded in the year 2000, and the Kohala Center is a nonprofit and a community of practitioners working to strengthen relationship with the Aina, to work knowing that when pilina is strong, our relationships are being cared for and fed. And if you want to learn more about them, please check out their website. They do a lot of amazing things. And as far as my project, this year I was working on assisting the crew with sweeping 300 acres of land per year to remove the invasive ginger in the forest. I was also helping in maintaining fence lines for our Eke unit and our Lapahoihoi Nui unit, as well as doing surveys in the forest, as well as our streams, and assisting in removing ungulates in our now fenced-in area. So I want to take you guys a little tour of where I've been working at. So, of course, I've been working in this lovely Kohala Mountain up in the cloud forest. But before we go up the mountain, we'll start in Waimea. This is where we all gather each day. This is where our office and base yard is. We do our meetings there, gather room into the truck vehicles to head up the mountain. And a little bit up the mountain, this is our Kauai corridor. It is a little Kauai Chi sanctuary that we help maintain. We do our fence line checks. This is actually where I did my first day of work. I was assisting in the treatment of fountain grass in the area, since it is an invasive species harming the plants there. And it was just an amazing place to start my work at. Moving on to our Eke unit, this is a part of the cloud forest that we are allowed to help take care of. It is where we mainly work. This is where we do a lot of our invasive ginger control and ungulate control. We also help monitor the forest up here, as well as our stream monitoring with our Keawe Vai stream up here, and a smaller stream down here that usually doesn't show up on maps. We also maintain the fence line and are trying to remove the ungulates in the area as well. And it's just such a beautiful forest to be able to work in this summer. Large trees and good side elevation we're at. And then another spot I was fortunate enough to work at was our Lapahoihoi Nui unit that we are contracted to help maintain. It is a remote area, so we had to be helicoptered in. And we also did things similar to our Eke unit where we are helping with ginger removal as well as doing fence checks, and I got the chance to camp up there. And up there had some really beautiful plants with their really red licos. And the ground was a little bit squishy with the moisture from the week before, but we had beautiful weather this week, so I was really fortunate for that. And then moving on to how I had to prepare for this, I had to learn a little bit about the land up here. I kind of knew about it before, but not too much. So this area outlined in the light green is owned by the Queen Emma Foundation. And you can see a noticeable break in the green vegetation. This upper part is what's part of the forest that we help maintain. The forest used to reach down further down Kohala Mountain, so we are helping to maintain what is still there. And this bottom section is what has been converted into pasture land by human activities and invasive species. And of course, before you start your workday, you need to understand where you're working at. We go to different areas every day, so being able to kilo is very important, not only for finding the target species you're looking for, as well as invasive and rare species, and as well as just being really safe, because this terrain is not for the faint of heart. You go up and down trees every day, you crawl under bushes, traverse aluhe patches, which is really fun. And just being aware of everything just makes everything much easier and safer. And of course, getting into the actual work, a lot of what we did is ginger control. So we were actually going through the forest, searching for ginger patches. As you can see in this video right here is how we cut the ginger. And we cut it below the last leaf to ensure that it can't photosynthesize. And then after all the stalks are cut, we do end up spraying it with herbicide to kill it right then and there, since it is harming our forest. This is also a new project. Since our unit is fenced in fully, we are doing some feral pig removal. We have traps set up in the unit that we bait, and we are trying to remove all the pigs within the area. And with these goals in mind, we hope that by removing and killing the ginger that is choking out our native species, we hope that we can allow native vegetation to come take over. We have a lot of ohia trees in the area that are at risk of rot since it is present in our forest. So we are doing whatever we can to help these trees survive for the long run. And by removing the pigs, it also helps since the pigs are able to eat the seeds of native plants, eat the seedlings, and they can help spread rot by causing injuries in the trees. And we hope that by these efforts, we're able to help this forest flourish. And then more about our ginger sweeps, it is really important that we have a lot of hands. We have a lot of acres we need to cover. If we're lucky, we get at least 2 acres a day since this forest is so dense. And we use GPS devices to track where we are covering so that we can put it on our database to show that we are actually doing the work and stuff is being done for our funding. And then our transect surveys, they're very important so we can help monitor the health of our forest. We do ungulate presence surveys to see if there's ungulates present in the area. We document native and non-native plant diversity, as well as document native and non-native bird diversity. And that's through our several transects. We also conduct stream monitoring to help monitor the health of our streams. Our main stream we cover is Kīhoʻihoʻi Stream, which does flow down into the ocean. So we're making sure that what is going down into our kai is healthy and not being overpopulated with particulates of any sort. And we also monitor another stream that we've dubbed KS2, which is just another stream in our area we monitor. And we do depth monitoring, flow monitoring, as well as turbidity and total suspended solids. Both are used to observe the water clarity as well as the amount of solids in the water for the stream's overall health. And I started this internship with a realized love for caring for the Aina. I think all my crazy stories of going up to the mountains or down at the beach have helped me realize that I really love working on this Aina. And as a major in environmental science, I have came into my host organization with useful background information that allowed me to jump into learning all new information and just loving the forest that I'm working in. While I felt that I have not been working at the Koala Center for that long, I hope that the work I've done has assisted and made some sort of impact in the future health of the forest. Even though I joined the crew at such a busy time with an acreage deadline due at the end of June, I hope that being an extra person in the ginger sweep line has helped make work a little bit easier for everyone. During the summer, I have found out this is the type of work that I want to be doing for the future. I have enjoyed working with everyone at TKC and walking in the forest, no matter the weather challenges, learning new skills, new species, and just gaining confidence in my everyday life. I learned that while this may be hard work, it is good work, not only for the Aina, but for yourself. I hope that I can one day return to the land that I worked in this summer. And once again, I want to thank the TKC field crew, Brooks, Jake, Ben, PE, Jay, and Megan. It has been an amazing experience. They have welcomed me into a crew without second thoughts and have taught me so much. And even in times where work got tough and the hike was tiring, they encouraged me to keep going. It was such a pleasure to work with them, and I hope to come back one day. Also, mahalo to all these sponsors that made this summer possible. Mahalo.
Mahalo nui, Jay, for just highlighting how great Kohala Mauna is. Do we have any questions in the audience for Jay, either online or in person?
Hi, Jay. Amazing presentation. Do you happen to know what the name of the pilot for the helicopter who rises? I might know who that is, and my mentor list is watching, and thinking his name is David Okita.
It is David Okita.
Oh my
Amazing
God.
guy.
Yeah, we all used to fly with him, and I'm great that you fly with him because he's like super safe. He's super amazing, and yeah, I just wanted to let you know that David is thebomb.com.
Yeah, mahalo David for safe flights.
Mahalo.
Any other Nino for Jay?
Aloha, mahalo for your presentation. I'm just wondering what has inspired you the most throughout your experience? Is it a place? Is it a hana, a certain day, or a mo'olelo? But one that you felt like really was the turning point in your summer?
I think the major turning point in my summer was our first Eke transect. We had the 5 of us going out on the transect, and it was raining so much the night before, so the streams just running so much, and like our bog that we had to go through was just like flooded. So at some point we just stopped like caring about staying dry and just like started just going through the puddles and just like having fun with it. We were all cracking up by the end of it, and I think that was really what like inspired me to want to do this more.
Awesome, mahalo.
Yes, one last round of applause for Jay, please. Oh, Kalamai, there was one more Nino. Mahea.
And I'm sorry to ask this very unpopular question, but since we're on the topic of aloha aina, how do you personally feel about the use of herbicides in the watershed in Wahokua?
That's a very good point to bring up. Well, I think that is such like a disastrous thing. I think that at some point it is necessary to help with the forest, because if we weren't using herbicides, I think we would have to be digging up the roots of the ginger, which I think could cause more harm in our forest. And I believe the crew at TKC they've done the research to make sure that what we're using is like targeted into the ginger stalks, so it's not at a great risk to harm the plants around it. And we do try to be mindful when we spray. We try not to spray into the water, and we try to just like target the stalks that we're spraying instead of just like taking a sprayer and like spraying everything in the forest. So I think they're trying to be very mindful of that.
Mahalo, Jay. One more round of applause for Kohala. And our last presentation before lunch will bring us back to this moku of Hilo down in Keaukaha. Miss Mika Gallardo will talk about the work that she's done with Hui Ho'ole Maluo. Kalamai, if you go back and you hit F11, it'll make the top part disappear. Oh, mahalo.
Okay. Aloha kako. My name is Mika Gallardo, and this summer I was placed with Hui Ho'ole Maluo. They're right there in the row. I don't know what row it is, but they're right there. This summer I focused on keiki curriculum based on the NGSS standards. To begin, I would like to start off with my hua. I'm originally from Mililani and Kamukio, Oahu. My academic journey started me off with my tropical forest ecosystem and agroforestry AA degree at Hawaii Community College, as well as my Hawaii lifestyles with the hula emphasis. The Hawaii lifestyles with the emphasis in hula. Throughout my academic journey, I was able to have a lot of internships that inspired me to be the person I am today. I got to work in a lot of different places. One of the biggest influences I had was my first job on this island, which was at the University of Hawaii's agriculture farm. I worked with the animal science department. This is where I gained my knowledge on different places around Hawai'i Island by working with the Uncle Ryan and Uncle Okimoto. They inspired, or we did a lot of branding and ranching, so I got to travel around the island, which influenced how I saw ungulate's impact on the island. So led me into reforestation, which led me into wanting to know a lot of things about the whole ecosystem. I recently graduated from the University of Hawaii at Hilo with my geography, environmental studies, and I really live my life by the Olelo no E'ao, Ohe Pau Ka'ike Halau Ho'okahi. Enough about me. I want to introduce Hui Ho'ole Maluo. Hui Ho'ole Maluo is an organization focused on the restoration and rehabilitation of local ia in the Ahupua of Waiakea on the southeast side of Hawaii Island. They were established in 2014, and they were officially named a nonprofit in 2019. They primarily focus on outdoor education and opportunities for organized community spaces and events with the focus in STEM, which is science, technology, engineering, arts, and mathematics. If you want to learn more about them, there's a link below. So my mentors this summer was mainly Elika Jardine, Kamala, Manoa, and Nahoku. Yeah, if you guys know them, you guys know that they're amazing people. The vision for Hui Ho'ole Maluo is a thriving community through thriving ecosystems. In my speech later on, you'll see how. Their mission is to advance our community by providing opportunities for Kupaʻaina to engage and reconnect with sustainable ancestral practices. So an example that I learned about this summer is, Kaumeke Ka'el likes to make a lot of their lunches with stuff from the Aina. So one of the meals that they like to make is miso soup with the limu that they find in the area. They name it miso ho'oheno, which comes from the Kaulu Vehio Kekai inspired, which was written by Auntie Edith Kanakaole. My current career pathway has led me on the journey of wanting to become a teacher in education. Yeah. So PIPES 2026 cohort has allowed me to dive deeper into that wanting to be a teacher through the opportunity where I create a curriculum for keiki that involves makahana ka'ike in correlation with the Department of Education standards. Just a little bit of information about sense of place. We're gonna go to Keaukaha. Three of the main sites that Hui Ho'ole Maluo takes care of is Honokea, Kaumaui, and Keaukea. These places in Keaukaha have different examples of fish ponds. If you're familiar with Honokea, raise your hand if you've been to Honokea before. Nice. Do you guys know another name for Honokea? Richardson's. Okay. Why do you think it's named Richardson's? Which guy? Yeah. Yeah, so knowing place names, you kind of learn the history behind why places are called what. So Richardson, or Honokea, it was home to the Ohana Malo. So the Ohana Malo asked Dr. Richardson's for help during an epidemic in the Keaukaha community. Because Mr. Richardson was so helpful for the Ohana, they gave him a parcel of land, which is now what we know as Richardson's today. But it's always good to know the old names or the other names of places just to have the history before all of those things that had happened. My ha'alele, or the process that I went through, is to create a well-structured curriculum package that supports learner success and possible future programs for hui ho'olemalu and personal use in my own career. To start off, I went through the Next Generation Science Standards, which was in the title, the NGSS, and looked through all of the, standards that a teacher would need to cover throughout the year. I started off by using the 5E instructional model: engage, explore, explain, elaborate, and evaluate. So through these different standards, I created lesson plans. So one of the examples of a lesson plan that I wanted to create, I didn't really focus on local year stuff yet because we didn't really dive into it. So I wanted to have the opportunity to learn. The standard that I chose was to plan and conduct an investigation to describe and classify different kinds of materials. So I would ask the students to just look through all the materials that I would bring and to use words to describe them. Then I structured an activity which they would kind of use their own mana'o to finally come out with a plan to make their own something. So they would choose between a beach picnic mat, a fishing basket, shade or shelter, garden tool, or rain hat, and use the materials that they observed and classified with their own knowledge, getting them used to what they see and the resources available which surrounds them. Our huaka'i involves papahana, puakahinano, which is a 1-week summer program for keiki, but it was 2 weeks because we had two different groups. Where they can learn either loko'ia, holokai, or hana no eao, olena. My focus was loko'ia, so our keiki was able to be exposed to different types of technology in which they can develop their own kilo observations and pilina. Together, they were able to work with the Mega Lab, the other local ias like Haleolono and Kumuola to learn different ways these places see their aina through different tools. So this is an example of the Mega Lab where they're building 3D models. We used it to build 3D models of the umu, which is located at Honokea. So the kids were able to, at first learn about how to do it on land, and then they would build 3D models of their peers, and then after they would go into the water and look at the umu. They also were able to use materials around them to figure out how to create a lens to look through the water, like goggles or like a camera. The goal was for them to see like how does looking through a camera make it different than just looking from above the water. Through this program, it was really emphasized that every keiki is different, every class is different. You can't treat one event the same as others, so you really have to adapt your way of teaching or to the styles that is the best for the group that you're with. Some kids or even adults need extra help because of their physical capabilities, maybe the amount of academic capability they have, or the progress in which they feel comfortable in an area. Sometimes the hardest thing to influence is a good mindset, so having people to be able to be open to try new things and learn about things through a different perspective. Another program that happened this summer was Kilo to Camera, which was more of a PD for adults and practitioners in the area. Kilo to Camera was also a 2-week program where we invited people in the Keaukaha community to collaborate on better ways that everyone can take care of their aina. We had people from Blue Technology, Robotics, to Lokelani Brand, to even other Kumuola, other teachers from Ka'umeke Ka'eo. We had Kipuka and Roxanne Stewart. Roxanne Stewart was she made one of the biggest impacts on how I see Kilo. She taught, the way that DOE kind of goes at Kilo now is just getting a sense of feeling and make sure the kids are looking at the environment, not really focused on a centralized idea. She taught us that Kilo is like, you have to have a focus and you can't just go out to an area. Like if you would want to look, let's say at Limu or something, you would, an activity she showed us was to look at Limu 3 different ways. Another Kilo practice she explained about was an Adventures of Master Aku'u, which she had taken pictures and had us interpret it through non-objective lens. So we had to make sure that there was like, we're inferencing they're doing this, and we just had to make sure that Kilo is totally just non-objective. Wendy Hamane is a Hilo high school teacher with the Lana Kilo Learning Center. She influenced my thought process about curriculum through multidisciplinary activities, like learning math through the removal of limu in the local and trying to have the kids determine how much of the limu they think is left within the pond based on how much was removed. She also does IMO research projects with the kids through the growth of kalo and other agriculture plants on the property. At the end of Kilo to Camera, we had different groups. I was part of the educational group. Through the educational group, we discussed recommendations that Hui Ho'ole Maluo can implement within their program. It ranges from extended learning, internships, research, community, schools, outreach, which we came up with 2 strategies, one internal and one external. An internal strategy for the program is to create lessons plans, which the program can use within their papahanas that happen throughout the year, where we would create like curriculum based on the core content standards and somehow relate it to place-based learning. For this example, one of my co-interns, she came up with an engineering and design project that talks about the kuapa and makaha. For our external strategies, we talked about professional development and opportunities, especially for teachers, how to get them involved within learning these processes to implement them into their curriculum in other schools around. In Keaukaha and around Keaukaha, this professional development would allow PD credit for the teachers, so a give back and reciprocation between the teachers and the site hosting. And at the end, we had an overview of how stewardship through education can look like a trusted educational resource, a catalyst for stronger school and community partnerships, a place that nourishes both people and place, a center for learning and practice, and a place that will feed its kaiaulu. Another event that we went to was Conservation Conference in 2026, is the Imi Ke'eweo Ka'aina. Through this conference, one of the main talks that kind of revolved around, I think, everything that we focused on this summer was a Paipaio Waikolu, which is a community science education group through Iolani schools that promoted research through their streams. So all the different schools around the area in Honolulu would come together and create a database and establish different ways they can kilo and make the streams around them better. And the last program that really hit home was the DOE first year teacher cohort. Through this teacher cohort, I was able to see how teachers right now are implementing what they're learning at place-based educational sites into their classroom. They talked about ha, or breath, which is the sense of belonging, responsibility, excellence, aloha, and total well-being, and Hawaiʻi. Just a kind of framework to make sure you would step into a classroom and feel like you're in a classroom in Hawaiʻi. It was surprising to me that not a lot of the teachers there felt really confident. in what they can bring to the table, but then we have to remember that everybody has their own sense of place of Hawaii, and that they just want to make it safe for like, I don't know, not a lot of people, like the safe space for people to feel that they have Ike and Hawaii stuff is being explored within our classrooms for DOE. So for my hoina, I overall learned that you need an entry-level hook. So something that grabs the attention of the keiki to want to learn more about what's going on in their environment. I also felt like I was successful in the reciprocity to give back to Hui Ho'ole Maluo and the Kaiaulu community of Keaukaha and the greater Hawaii Island. My overall project included making a pre-test for keiki. So this is where you would give them before they would come to the program. So you kind of want to know if they know a little bit about what's going on. And then we would go through a curriculum and try to explain a little bit more about what we want them to learn. So this workbook includes just like, you want to facilitate the learning and not really tell them what they want to learn. You want them to ask more questions and just to have an opinion about what they want to learn about. And then a post-test to see if your overall goal was met. For my ha'ina and future endeavors, I was just recently hired as a second grade teacher at a public charter-based school for place-based learning, and I want to implement this. I want to implement this curriculum into their first quarter project. So a give back would be bringing them back to Hui Ho'ole Maluo and using the curriculum that was built this summer, which is in the plans, which I'm excited about. I just want to mahalo the crew again. Manoa, Nahoku, Elika, and Kamala, along with Hulali P and Hulali W. Mahalo nui for this summer. And Cody Faye for all the beautiful photography. Any questions?
Oh, plenty questions for Mika.
Oh, mahalo, Mika. Mahalo for jumping into a summer of unknown and brand new things. I have a twofold question. One, you talked about your journey through college and forestry and ungulate research and then geography. So why Lokoi'a and why coastline communities like Keaukaha for the summer? And then two, what are like top two things that you're taking from, because we went through programming, right? We went through programming with keiki, with adults, with all these things. Okay. Two things that you're taking from those curriculums and from those programmings into your classroom this next year.
Okay. So wait, what was the first question?
Why Lokoi'a?
Oh, why Lokoi'a? Because I'm not comfortable in the water. So I figured out that because I was so much in the mountain, and the point of reforestation is to work with the water cycle and just gaining water back. I just wanted to see how everything that happens in Mauka can relate to everything that happens in the Kai. So why did I choose Lokoi'a? Because I'm uncomfortable with it. So mahalo for being patient with me. And the second question was two things that I'm taking away is one, that organization and coming into a student program is key, and knowing what's going on, which is something that I need to work on more. So that's where a lot of my personal growth happened, or is happening. And two is that you need to just go with the flow, because sometimes you think your plan is going to work out one way and you need to be able to be flexible. Yeah. Mahalo.
Aloha. Thank you so much for your presentation. I wanted to ask what age group you worked with, and then if you had examples of like your favorite moments when you were teaching them what to do.
Okay. So we were working with K through 6, and an example of something cool that happened was that this student who really wasn't comfortable or confident in his swimming skills. So throughout the weeks, he was able to jump in the local through all three of them, starting with Honokea, and then we went to the ones on the Kaumaui property. And throughout the progression of the weeks, you could see how he was able to swim on his own with a floaty device and put his head in the water. So he was able to look at the fish by himself. Another one would be, I think he was kindergarten, sorry. Another one would be, I think a fourth grader. He felt more confident in what he knew, and he was able to take on leadership roles. It was cool to see within the students their own confidence being built. And when when the technology part would come up, the kids were really interested because it looks like robots and what is it called? Or it is robots, but it looked like video games. So they were cool how to apply those video game looking styles into what's actually happening in the place around them, which made them more interested in what's going on in the water. I thought that was really cool. We had a device that was controlled by like a Xbox controller kind of looking thing. And then the kids really were into seeing how it moves around and all that jazz. Yeah.
We have one question online from Uoloku, and they ask, how has this experience influenced your master's degree pathway? I know you just finished school, but what's next for you?
Okay, so it took me forever to get my bachelor's, yeah. But I really want to go back for my master's in education. I'm not sure what program I want to go with because there is the one at UH that's really good, and I took a lot of classes there as well. But then there's also the charter school master's program. I'm not sure what it is. Kohoʻiwai? Kawaiola? Yeah, one of those. Kohoʻiwai, yeah. So it really influenced my wanting to go back for education. Especially, yeah. Mahalo.
Let us know when you do, and let us know when you have your next Pukakula so we can all be there with you.
Aww.
So can we get one last round of applause for Miss Mika, please?
Mahalo.
And that concludes the first half of our morning. We have mea'ai, we have lunch that is on its way, hopefully very, very soon. But if we could all make a po'ai and pule so that as soon as the food comes, we can grind.
I just got handed the mic. So Kiana will be back, I promise. We are going to start off our last bit of our presentations for the year. No pressure, cool. We're gonna learn all about what you've been doing. So please go ahead.
Aloha mai kākou. No leila o wau no o ka hokuala Kwanan I ke kauwela nei ua kālele au ma kanana ana I nā palapala mahele ma kona I mea e hiki ai a mākou ke kahai palapala aina o nā wahimahi ai. Ua kapa inoa wau I ke aho ike o ka hoʻonā ia o nā kona. Ka hoʻohana ana I nā palapala mai ka wā mamua I mea e mahi pono ai I ka wā ma hope ma Hawaiʻi nei. No leila, I mea e hoʻomaka mai io lilo loa mai, makamaka hiki 18, kana 5.3, ma kauhako a me hoʻokena ahupuaʻa, ua haʻawi ia ka ʻaina kuleana I koʻu kupuna kua mea lā. O kona inoa Elia Kauē, kekahi mau makahiki ma hope mai, ua hanau ia koʻu makua kane a hanai ia ma ka awaloa ma kona hema. A leila, ma ia wā like ua hanau a hanai ia koʻu makua hine ma Hilo nei. A leila, hui pulawa, a leila, hanau mai o ka hokuala Kwanan, a ua hanai ia wau ka nui o ka manawa ma ka hawa leʻa puna. He ahupuaʻa ma kahi kokoke o hale maʻu. Ua hoʻomaka koʻu huakaʻi I mina oau o lalo Hawaiʻi ma ke kula o nā wahi o Kalani o Puʻu. Ua hoʻo ia wau ma ia kula ma koʻu wā papa malaʻo a I koʻu pukana. A lila ua hoʻomau ia no ma ka makahiki 2.2 ma ka hakaʻula o ke ʻelikolani ma keia kula nui nei. He me kia pālua wau ma ka haʻawina Hawaiʻi a me ke kālai politika. Oia ke kumu, ua ike wau I ka ʻōlelo Hawaiʻi, ua aʻo wau I ke koʻikoʻi o ka ʻōlelo ma ka mākou noiʻi ʻana. I kēia kauwela, ua hana wau maʻaneʻi nei ma kēia hale. Ua hana wau ma ka Indigenous Cropping Systems Lab, ai ʻole ke keʻena mahi ai o iwi ma lalo o ke kulanui o Hawaiʻi ma Manoa. I mea e hana ai ma kēia pahana. No leila, o ka pahuhopu nui o ka mākou pahana, oia ke kukulu ʻana ma luna o ka Marian Kele Puke, kapa ia o nā mala o kona. Ma kana noiʻi ʻana, ua noiʻi oia no nā wahi mahi ai ma kona, oia nā cropping zones. Leila, ua hoʻohana oia I nā palapala mahele I mea I ʻimi ai I kēia mau ike. No leila, ua loaʻa e ke kahua no kēia pahana a ke kukulu wale nei mākou ma luna. I mea e ike mua kaka ai kākou I kēia mea o ka mahele, e wehewehe lihi ana wau I ka mau aukala o kēia hanana. No leila, ma ka makahiki 18 kanahā, ua hoʻano hou o kaui keuli I ka palapala pono kanaka. Ma kēia palapala nei, ua ai oia I ka ʻaina alodio, oia no hoʻi ka ʻaina I ona ia e kekahi kanaka. No ka manawa mua ma Hawaiʻi, a leila ma ka makahiki 18 kanahā kumaono, ua hoʻomaka ka hoʻomahele ia o ka ʻaina, ai ʻole ka mahele. Ua hoʻomaka me nā ʻaina kalaunu, aupuni, a leila nā ʻaina konohiki a me nā ʻaina aliʻi. A leila ma ka makahiki 18 kana 5, ua ai ia nā maka ʻainana e hoʻouna I kekahi kainoa no kā lākou ʻaina ponoʻi, oia no nā ʻaina kuleana. No ka hoʻokainoa ʻana I ka ʻaina penei ke kaʻina hana, o ke keʻehina mua, oia ka hoʻouna ʻana I ka hoʻokainoa ʻaina, oia no hoʻi ka register. O kia kekahi la'ana, o lelo ia, eia ko'u wahi kuleana ʻaina o Pahua Ka'ili, ma loko o ka lā hiki ke ahu pua'a. He eiwa ki hapai kalo, ho'okahi ki hapai uala, a he ho'okahi ki hapai ohe. No leila o keia ka ma'amau no nā ho'okainoa ʻaina, o wehe ke kanaka nana ka ʻaina, he ahana mea ana e kanu nei, a me kekahi mau inoa wahi kekahi. O ke keʻehina ʻelua, o ia ka ho'ouna ʻana i ka palapala ho'ike, ai ʻole ka testimony. O ka ma'amau mai kekahi hoʻakaia home keia, ai ʻole kekahi mau hoʻakaia home i mea e ho'oia ai a pono ai i ka mea i ʻōlelo ia e ke kanaka nono ka ʻaina. No leila, maneʻi nei ʻōlelo ia hoʻohiki ia ua ʻike māua i kona ʻapana ʻili o Pahua ma ka lā hiki. No leila, o keia he hoʻoia wale no i kāna i ʻōlelo ai. A leila o ka hope o ia ka haʻawina ʻaina, loa keia palapala nei o ia kekahi la'ana o ka haʻawi ʻana o ka ʻaina, e naʻe ka hapanui o ka manawa ua hoʻole ia ka ʻaina, a me ka haʻawi ia o ka ʻaina i nā makaʻainana. Ma hope o ka nana ia e ka luna hoʻonā kuleana ʻaina, o ia ka Board of Commissioners to Quiet Land Titles. Maʻaneʻi nei, wehe ia nā palena, a kekahi mau manawa loa nā mea kanu ma luna. A ka hapanui o ka manawa ʻaʻole loa kēlā i ke pili. No leila, o ka maʻamau no ka mākou hana, ua ʻike mua ʻoukou ua kokua wau ma nā pahana e aʻe ma ke keʻena. E naʻe no keia pahana, ka hapanui o ka manawa aia mākou ma nā kamepiula, hui mākou ma ka Zoom o iai noho o Tami ma kona i kia manawa. Hana mākou i nā mea noiʻi, hoʻoia i koʻu mau hana. Hana mākou i nā mea palapala ʻaina ma GIS. O keia, kekahi ʻapana o ka pakuhi nui a māua i hoʻokumu ai, loa nā ike pili a pau no nā ʻaina. Lehi ke ike maʻaneʻi ka heluna haʻawi ʻaina o ia ka LCA. A leila ke ahu puaʻa. A leila e hia ʻapana i haʻawi ia, a me e hia ʻapana i noi ia. O ia ka hapanui o ka manawa, ua hoʻole ia ka hapanui. A leila kekahi mau kākaha. Maʻaneʻi nei kekahi lāʻana o ka hoʻokainoa ʻaina a koʻu kupuna o Elia. O ka maʻamau helu wau i keia mau mea a ʻano paʻakiki o he limahio, Hawaiʻi. E naʻe, wehe lākou i ka lākou ʻaina, kekahi mau manawa, ka lākou hana ma luna. Ma kea mea kikoʻī wehe ʻoia i kona pahale ma kai, a me kona lawaiʻa ʻana, a ma hea kahi e lawaiʻa ai ma kauhako. O kia kekahi mau i ke pili heluna, ma kona he walu haneli kanaha kumalima mau heluna haʻawi ʻaina. Leila he makahi o ka ʻelua kaukani, ʻelima haneli iwakalu kumaiwa mau ʻapana i noi ia. A leila hoʻole ia ka hapanui. A leila haʻawi ia he ʻumikumalua haneli. He hoʻokahi haneli kana walu kumalua i kaha ʻole ia ma kekahi palapala ʻaina. A leila loa he hoʻokahi kaukani kanaha kumakahi i koi. E kolu haneli kana walu he mau pahale. O ka maʻamau ʻaʻohe mea e mahi ia nei ma ia mau ʻaina. A leila e ona haneli kanono kumakahi mau ʻapana ʻaina. A leila e hā haneli e kahi mau ʻapana me ka mea kanu i ʻōlelo ia. O ia kekahi mau manawa hoʻouna ka mea nana ka ʻaina i kekahi hoʻokainoa ʻaina. ʻAʻole naʻe ʻo ia ʻōlelo i ke ʻano o nā mea kanu ma leila. A leila e ono ʻapana me ka wahi mahi ai, ai ʻole ke cropping zone i ʻōlelo ia. No leila, o kia kekahi lāʻana o ka palapala ʻaina i haku ia me nā ʻaina a pau i loaʻa i nā makaʻainana o kona. Nā Tami kēia, yesa. A leila, o kia kekahi kiʻi mai nā makahiki ʻumikumawalu ʻiwakalua. Ua kahiki ia mai mai kai o kailua. He mea ano shmio keia o e hiki ke ike me ka moa kaka I nā mahele mahi ai e like me nā kumu ulu ma ana I nei. No leila o nā mahele mahi ai o kona, o ia ke kula, ke kalu ulu, ai ole ka ulu belt, o ia ke kahi noa kaulana. Ka apa'a, o ia ma kahi o ka halu'u kuahewa. A leila ka ama'u, a o keia kahi mauka loa I mahi ia e nā kanaka ma kona. Me keia ike pili, ua hiki a mākou ke haku I keia palapala ʻaina. A hiki ke ike maʻaneʻi o ka mele mele o I ke kula, o ka ʻōmaʻomao o I ke kalu ulu, a leila o keia ka apa'a, a ma'u, keia ma mea uliuli o ia ka pai keala ehu. No leila o keia nā wahi mahi ai a pau a mākou I imi ai, ke kula ka paheʻe nā maua I imi, o iai ʻaʻohe kanaka I noiʻi no keia wahi. Kalu ulu a pa'a a leila keala ehu, o iai ke kahi wahi a maua I imi ai, a leila ka ama'u. No leila o keia ke kahi hana e aʻe I hiki ai a mākou ke hana ai, me keia ike pili o ia ka haku ʻana I nā palapala ʻaina I pili I ke kahi mea kanu kikoʻi. No leila o keia ka palapala ʻaina no ka ʻalani, o keia ka palapala ʻaina no ka maiʻa, o keia ka palapala ʻaina no ke kope, o keia ka palapala ʻaina no ka uala, nui ka uala. A leila o ka mea nui loa a mākou I ike ai, o ia ke kālo. Yes, sir. O keia ke kahi mau heluna I ke pili e aʻe, o keia nā mea kanu I ike nui ia ma kona, a hiki ke ike ke kope a me ka ʻalani, a ole o ke la he mau mea kanu o iwi, e naʻe ma ka wā o ka mahele ua loʻa e ma Hawaiʻi ai, no leila ua kanu nui ia. A o keia ke kahi mau mea kanu e aʻe a mākou I ike ai, he mea ʻano hoi hoi, o iai o ke kahi koina I hiki ke loaʻa I ka ʻaina kuleana, o ia ka pono e mahi I ke kahi mea ai I hiki ke ʻai ia. E naʻe ua loaʻa ka mea, ua loaʻa nā ʻaina me ka hala, ke kou, ka hau, a me ka loulu ma luna, nā kumulaʻau o ka maʻamau a ʻole I ʻai nui ia. E naʻe ua loaʻa ka ʻaina I keia mau kanaka. No leila, ma koʻu noʻonoʻo ʻana I keia kauwela I halaiʻo nei, ua kau nui koʻu manaʻo ma keia ʻolelo eau o I ka wā mamua, ka wā mahope, o iai me ke aho wale, ke hoʻi nei mākou I hope I mea e nānā ai I keia mau palapala. I hiki a mākou ke mahi pono, a I hiki a mākou ke mahi I nā mea ai I pono no mākou, a I pono no ka ʻaina. Ua ike leʻa mākou, ua hana mua ka mākou mau kupuna I keia mau hana, no leila he mea nui ka hiki ke hoʻola I keia mau ike mai ka wā mamua. O keia ke kahi ʻolelo no e au aʻui noʻonoʻo nui ai, o ka ʻolelo no Keola I ka ʻolelo no Kamake. Pili kia I ka ʻolelo Hawaiʻi, ma ka ʻolelo ʻana a me ka hoʻohana ʻana I ka ʻolelo Hawaiʻi, he ola ma leila. I loko o keia noiʻi ʻana, ua ike wau I ke koʻikoʻi, o iai I nā ʻaʻohe kanaka I hiki ke ʻolelo Hawaiʻi, a helu helu I keia mau palapala, a ʻole I hiki a mākou ke unuhi I keia ike. No leila, he mea nui ka mākou hoʻohana ʻana a me ke aʻo ʻana I ka ʻolelo Hawaiʻi. I hiki ke hoʻomau I keia ʻano noiʻi ʻana. O ka mākou pahu hopu, o ia ka hoʻomau ʻana ma nā ʻaina e aʻe, a ma nā moku e aʻe no paha ma kia moku puni, nā moku puni e aʻe, a me nā hui o iwi e aʻe a puni o ka honua. He mahalo nui kēia I ke keʻena mahi ai o iwi, o ua nui nā mea ui a oi, ma waho o ka nui I ana ma nā palapala mahele. O ua kokua wau me nā kanaka e ae. Mahalo ia Sema me Kimo no ka la wahana pū ana me ia ui keia kauwela o ua le'a no. Mahalo ia ho'oulu la hui no kona ho'oia mau ana ia mākou. Mahalo ia Noah Lincoln o kana alaka'i ana. Mahalo ia Jacob Hurst. A leila mahalo nui ia Tommy Hensel o ia nana no I hana I ka nui o keia pahana. A nana no I ho'omaka a e ho'omau ana o ia ma ka wai hiki mai ana. A he mahalo nui kia ia oha o iai wahana pū mākou me lākou, o iai nā lākou nā palapala a mākou I nāna ai, no leila mahalo nui a lākou kekahi. Mahalo!
Nino. Kaliiki.
Sup, cool.
Sup, bro?
Love you, bro. Nice presentation. How would you say this summer project has perhaps changed or maybe given you some new ideas for what you want to do as a recent graduate or like your future professional trajectory?
Hiki ke panema ko pelikanio? Oh, okay. So basically, don't worry guys, I can speak English. I think this project has really helped me a lot in like my organizational skills, and I think that'll be really important when I go into grad school. And it also taught me the importance of keeping Olelo alive, because like I said, if no one was around to Olelo or read these documents, there's really no way that we could get all this information. So I would definitely want to incorporate more Olelo into my post-grad schooling. And yeah, mahalo.
Mahalo ku. Kanina a'u peheo ko e ike ai ka nui o ka hana ma ke la mau a pana aina, ma ke la palapala aina I hoi ke ia. Like, ka aina kalo, mahi ai kalo, ka aina mahi ai mai a pela aku.
Ike ma ka nui o nā aina, ai ole?
Like, ka hana. Pehe o koe ike ka hana ma ke la mau aina I ka waka iko.
O iai, e like me ka ui o lalo muai, ke ho'ouna, ke kanaka nona ka aina I ka ho'o kainoa aina, o ia ka register. Ka nui o ka manawa o lalo ke kanaka, ke kanune au I keia me keia. O ia ke kahi mea mai ka iloa o nā palapala mahele, o ia hiki mako ke ike kiko I ma ke kahi palapala aina mahea o ia I mahi ai.
Malo.
Malo.
Oh, he mau Nino. He nino ka'u ke kahi.
Aloha.
Aloha.
Oi, kalele nui oi, ike wau kalele nui oi ma luna o ka awa ho politika. Oi, ka hoi. Ma ke wau e niele ko manao no nā mea e like ai a o ko ai ke aloha aina a me ka malama aina. A hea nā mea e ko kako ana ke kahi.
No leila ke aloha aina, o e nino oi no ka au politika, he mea politika ke la ma ke kahi ano, o iai ma ka wakahiko o ke aloha aina, o ia nā kanaka I ka ko'o I ke kalaunu, a I kea mau la o ke aloha aina, o ia ke aloha ana I ka aina, ka hana ana ma ka aina, a pela wale aku, e nā e loa ke kahi mana'o ano malama aina ma ke ano aupuni a me nā kana wai e ho'oia ana I ka malama I ano ka mako aina, ko mako aina. I ko mana o oia ke ala e ka ko'o ai ke aloha aina I ka malama aina o iai me ka ole o ke aloha aina ma ke ano kanawai a ole I hiki a mākou ke malama I ka aina ma ka aina o iai a ohe aina e malama ai no leila pono I ke aloha aina no ka malama aina. Mahalo.
Aloha, mahalo nui no kau hana maika'i a hoi hoi. He nina o kau oiai he iini ko kekahi o mākou ma kona e kukulu I nā ho'ailona. E ho'ike ana I ke lame keia ahu pa'a, nai noa. E like me ko ahu. Ua maka mākou e hui a halawai no keia mama ho'olala. O kau nina o ia oi, he mana o ko kua paha, he a'o paha ko no mākou no ka ho'oia ana I nā wahi a me nai noa o ke ala me kia ahu pua'a.
I ko'u mana o ka mua, he nina maika'i ke la no Tommy Hensel. O ka lua, kekahi a'o I ko mana o he mea maika'i ka nana ana I nā palapala mahele o iai ua hiki a mākou ke ike keu ho'i o iai ua ho'ohana mākou I a Kipuka database kekahi mau manawa o nā palena I ho'okumu ia I kea mau la a ole I like me nā palena ma ka wā kahiko keu ho'i ma ka wā o ka mahele no leila kekahi la ana o keia o lalo ia ma nā palapala aina o kea la o aia keia wahi ma hea kekahi la ana o ma ho'okena e ina nana oe ina kanaka I noho ma leila mai ia wā o lalo lako o kiloa keia e ole kiloa e kahi, kiloa e lua no leila he mea mai ka I ka nana ana a leila ina makemake o koe nana ina ili kekahi o iai he nui a lehu nā ili maha kona o ia kau mea I ike ai no leila ua hiki ke ho'omaka me nā ahu pua'a ma ke ano he kahua ike a leila ho'omau ia la me nā apana e a'e.
A laila o ka'u ninau, ua ike paha oi kekahi mea pu'iwa, kekahi mea hoi loa, a ole paha oi mana o ike ana oi ke ia ma kekahi palapala, kekahi palapala aina, kekahi leka, kekahi nupepa, like kekahi mea pu'iwa au I ike ai.
O kekahi mea I ano ho'opu'iwa ia'u ma ke ano maika'i o ia ka ho'okainoa aina a ko'u kupuna o Elia o iai no nā pāhale a ole I kanu ia kekahi ano mea kanu ma leila no leila o ka hapanui o ka manawa ua ho'ole ia e na'e ma kona ho'okainoa aina wehe o ia a ole wau ke kanu nei I kekahi mea ma'ane'i e na'e he ohana lawai'a mākou. A manawa he mea ano hoi ke la o iai lawai'a mākou I kia manawa ma kahi like no leila o manawa ho. A ole wau I ike he mea he ike mai ka pō mai a ole wau I ike he ohana lawai'a ka mākou ko mākou mai. I o lilo loa mai ko hu mea la no leila. Ai oi kekahi mea ho'opu'iwa ia'u.
Mahalo. Can we get one more round of applause for Ku? And up next we have Mr. Ethan, who's gonna tell us all about our Manu friends up in Hakalau. Oh my ka'ie, Ethan!
Can everyone hear me? Yay. Okay. Aloha mai kako. Ethan Ko'inoha, and today we'll be listening to the forest, learning about Nā Manu Nā Hele o Hakalau, and I'm the 2026 PIPES intern with the Lohe Bioacoustics Lab. So I'll just jump right into it. For my hua, just a little bit about me. I like birds. I started birding around 10 years ago. Just my sister and I in front of the feeder every weekend at our local preserve in New Jersey as part of Cornell's Project Feeder Watch. And since then, they've kind of taken over like every single aspect of my life. I started birding more on my own throughout, especially during the pandemic. And then once high school started, that's kind of when I met my first like main ornithology, banding, skinning mentor. Her name is Hannah Southers, and she's from Maui. And she's one of the main reasons I wanted to apply for this program. She's turning 96 in a few months, and she's still going. So really exciting. I learned so much from her and all the other banders at the banding station. And the skills I learned there led me to my first really fully immersed tropical ecology experience in the Amazon rainforest, banding birds with Fauna Forever in Peru. But that's also where I first got exposed to kind of the heavier side of conservation that I'm sure we're all familiar with. We saw a lot of the illegal gold miners. We heard the bombing at night. There was a wildfire the size of Switzerland while we were there. But all of that kind of just strengthened my resolve to do the work that I do. And this is what I want to dedicate my life to. And I just finished my first year. I'm studying environmental science and data science at UC Berkeley, but all kind of oriented around birds. And now I'm here in Hawaii. And for all the bird banders out there, I organized this by the molt cycle aging codes for each bird. But if you're not familiar, first cycle alternate refers to your first flashy, gorgeous breeding plumage. So basically, I'm living my best life right now. Yep. I just want to say it's such an honor to be able to work with these birds. In the birding community, they're very highly revered. You're working with the Hawaiian honeycreepers. That's so amazing. The endemism, you can't find these birds anywhere else. The way so many of them are so specialized in each of their niches is just so special. So really an honor to be able to work with them. But unfortunately, Hawaii's also been dubbed the extinction capital of the world just because of the sheer number of extinctions of various species. There were originally 84 species of native Hawaiian forest birds, 58 of which are now extinct and 26 are left. Out of those, 59 were honeycreepers, 42 of which are now extinct and 17 are left. One of the main reasons is clearing of habitat for cattle ranching and other factors, but also the introduction of Culex cuncafasciatus, which is the southern house mosquito in the late 1800s, brought here by European and American sailors. And one of our most iconic species, the i'iwi, has a 90% average mortality rate, sometimes even higher for juveniles, for malaria. So a really huge threat. But I see myself fitting into the Lohe Lab's work as part of the story in place of Hawaiian birds, mainly as an apprentice to help leverage new technologies that improve long-term monitoring of bird populations and resulting management strategies. And just a little bit of background about where we're working. I was at the Hakalau Forest National Wildlife Refuge this summer. A little bit about Hakalau, it roughly translates to many perches. So some of that Ike Kupuna, very excellent habitat for native birds. The National Wildlife Refuge was established in 1985 to protect native Hawaiian birds and the remaining stands of native montane rainforest they depend on, as we notice these lower elevation populations collapsing. The Hakalau Forest Unit is along the slopes of Mauna Kea, pretty high elevation, 3,500 to 6,000 feet. And it's a mix of old-growth ohia and koa forests. And through outplanting by the U.S. Fish and Wildlife Service, much of the former cattle pasture is now restored koa forest. And we're seeing that a lot of our endangers really like this. So Alavi and Akiapola'au, you can often hear singing in them. And it's the best bird habitat left in all of Hawaii. It's one of the only places where native Hawaiian bird populations are stable or increasing. And it's also a really great place to see our three endangereds there, Alavi, Akiapola'au, and Akepa. And this is just a picture from the bottom of one of our transects, but you can just see like really like gorgeous habitat. Such an honor to be able to work in this space. And as Pat calls it, it's the last best place for Hawaiian birds. But unfortunately, with climate change, ecologists predict that warming conditions may allow mosquitoes to infiltrate these high elevation safe zones like Hakalau, especially with the super El Nino this year. Long-term monitoring of bird populations and avian malaria presence is really crucial for native Hawaiian bird conservation. And this is just kind of a figure that shows that as temperatures get warmer and warmer, mosquitoes can go higher and higher because the main thing keeping them out right now is the cold. And there's also a specific temperature at which they become infectious, like the actual plasmodium in their bodies. So yeah. And I think one of the Olelo no Eao really captures this well. It's Aohe kahua ona manu, or there's nowhere for the birds to land. But there's things that we can do to help fight this. That's the main reason why we launched the Ulu Manu project recently, which has 3 main objectives. Conduct passive acoustic monitoring to detect changes in bird abundance over time at Hakalau. Conduct oviposition sampling for mosquitoes along established transects, so just checking if there's mosquitoes there and if they have malaria. And to assist with restoration of ohia forests at high elevations. And this is a map of the transects that we do. And actually, these are not easy transects. But like since they're like kind of like hard to access, we're some of the only groups that are actually down there sampling. So really important knowledge gap that we're filling. And also just with passive acoustic monitoring, these new technologies, recorders, and like bioacoustic data analysis software is allowing us to maintain this like basically network of so many ears in the field 24/7 listening to these birds, which we couldn't really do before without these technologies. So we're hoping that this type of strategy can allow us to get a more complete picture of how their populations are doing and then how to better help them. So for my ha'alele, a little bit more about the methods. I started off by learning how to use Raven Pro software, which is a bioacoustics data analysis software for identifying and selecting birds within a soundscape. And we don't really use this as much for analysis anymore, but is mostly to get familiar with the native Hawaiian bird sounds by annotating past soundscape recordings. And this is what that kind of looks like, and that's what it sounds like. Yeah, so really beautiful sounds from the forest. But to maintain those recorders, you have to do a lot of field work. So the specific recorders that we use are song meters. So when we go out to the field, we just check if they're working, collect the SD cards that contain the actual recordings, and replace batteries and give them new SD cards. At each recorder, at certain recorders, we conduct oviposition sampling. So oviposition is just laying eggs. So we just put these cups filled with a pungent mosquito attractant that we call stinky water. It's just a mix of hay and yeast, but it releases carbon dioxide and attracts the mosquitoes. And we're focusing on the lower elevations because we're expecting the mosquitoes to move higher and higher up the mountain. So we want to catch them before they infiltrate the higher elevations. So we're putting them lower first. And we also do plant phenology surveys at each recorder, so just checking what species are around, are they fruiting or flowering, and does that correspond with any peaks in bird vocal activity. And these are some pictures from the field. These are not easy transects. It's a lot of overlook, underlook, in between the lot, but oh shoot, there's no carpet, down the riverbank, up the riverbank, climb the road. Even coming from the Amazon rainforest, I don't think I've ever been this wet, but it's also just so amazing to be barreling, charging through the forest and hearing all these native Hawaiian birds. Just really special place and experience. That's us maintain the recorders. That's an example of what the ova cups look like. So inside that is the stinky water. And along the way, we also look for hapu'u cavities, which are prime breeding ground for mosquitoes. We just check if they have any culex larvae, and egg rafts. Luckily, we haven't found any recently. We have found Aedes japonicus, but they don't carry avian malaria, so we're good so far. But this is kind of an early warning detection system. So if we notice any mosquito larvae that are culex or like there's a decline in native Hawaiian bird populations from the recorders, then we can kind of go into like BTI, IIT, eradicate the mosquitoes before they get higher. Yeah, thank you to the field crew. It's really fun up there, really special place. And then the main software we use now is Google Perch, which is a convolutional neural network that automatically detects the acoustic presence of a bird species within each 5-second window of the recording, and it gives us a confidence score. Yeah. So for my huaka'i, I helped a lot with the broader Ulumanu project, but within that, I was working on a vocal phonology study for native Hawaiian birds. So basically understanding when and also where birds are singing versus calling. And if you didn't know, birds' songs and calls are different. So birds' songs are like longer, more complex, and they're like, they can have different meanings. Like it's like, oh, this is my territory. Like, you don't come in my territory. If you come in my territory, we're gonna scrap and we're gonna have problems. Or like, oh, I'm so beautiful. Like, you should come like talk story with me and reproduce. That's bird songs. Then for calls, there's also a few different types. There's like alarm calls, like, oh shoot, there's a predator, we should run away. Or like, oh, there's contact calls, like, oh, here I am in the flock, and they're usually like shorter, like simpler. Yeah, really cool. We're hoping that this work will help streamline the U.S. Fish and Wildlife Service surveying protocols, because previous vocal phonology studies found significant discrepancies between the peaks of bird vocal activity, so what the birds were actually doing, and then what the experts were saying they were doing. And we also may investigate the impacts of environmental variables on bird behavior, which may also indicate underlying changes in their biology. So like, we usually expect birds to be singing more during their breeding season, because that's when they're reproducing. And just this whole experience like allowed me to get familiar with a lot of the parts of the scientific process. So literature review, looking through old papers, collecting bioacoustic data, and also analyzing that data in the lab. And this is just kind of one of the inspo studies. But you can see the dark blue is like where the experts said the birds were singing, and the dark orange is where the birds were actually singing. So there's like some pretty big like misalignments. So we're hoping that with our work we can help like calibrate what the surveying protocol is to what the birds are actually doing, so we can better detect them, know what their populations are, and then protect them. But before doing a song called vocal phonology study, I had to manually sort the existing training data set of 8,780 recordings based on the presence of bird songs and recalls. So in between the field, I was just plowing through 8,780 bird song recordings. So a few thousand for each species, and this is kind of what that looks like. Thank you, Amanda, for setting up the notebook. This one's for a kappa. I think they sound like fairies. Little D. Yeah. Yeah, and it goes on. But they're my favorite. I think they sound like fairies. After that, the notebook sends these recordings into these folders. And after that, we can train the model based on these manual classifications. And I'm now moving on to top-down validation of the model's automatic classifications to determine 95% and 99% precision thresholds. So basically, numbers above which the model is 95 or 99% accurate. Afterwards, we can calculate the detection rates and compare them across sites and time periods. That's just kind of what the model looks like behind the scenes. And these were the thresholds I got. So I listened to another 1,840 Akiapola hour recordings, and then we found that the most conservative threshold was actually from this year's breeding season. And you usually take the most conservative 95% threshold, but you can see that this 95% threshold is actually like closer or even higher than a lot of these 99% thresholds. So we're getting pretty accurate classifications here. And after that, we can plot threshold-based counts of songs and calls over time, observe any correlations with changing temperature and precipitation patterns from climate change, compare song phonology with breeding phonology, so that's usually determined by banding. CP is what the males use to talk story, and the BP is what the females develop when they like drop feathers along their breast to better transfer heat to their eggs when they're incubating. And then we might also create site-based song and call detection heat maps to see where in the refuge they're singing versus calling and when. And also dial seasonal heat maps, so when across the year and during the day, and detectability time series as shown before. And this is one of the graphs we got. It's really cool because there's an actual pattern. So you can see these like big like peaks around like the middle of July when you would expect them to be breeding, which is really cool because they actually breed at a different time than the other birds. So these results really reflect that well. And it's very consistent across years. Like you can kind of see the peak pretty much every year. And also across sites too. Like this was Puakala. And this is Pedro. And you can see this similar like peak pattern repeated. So really cool to see that. I started with Akiapola Owl because it's like probably like the least numerous out of all our birds that we have up there. But later on, like I'm in the process of going through a Kepa right now. Later I'll be going through like Gina's helping with Alavi, and then like Amakihi, Alapayo, Oma'o. Yeah. So for my ho'ina, I learned so much this summer. I learned to identify native Hawaiian birds by sound, maintain acoustic recorders for passive acoustic monitoring, use Raven and Perch for bioacoustic data analysis, conduct oviposition sampling for mosquitoes, and also learn the native plants for the plant phenology surveys. Also, like more informally, I got to see more of like the bureaucracy behind all the conservation work we do. Like involving access to funding and purchase orders. I'm sure we're all familiar with that. I think there was this one time Keely and I were dragging these like 9 like giant planks of wood for these seabird nest boxes. So just like taking all these amazing ideas we have in the lab and like transferring that to the field. This was from one of the last days at Hakalau, but I got to process an EEV. Really cool. For accomplishments and challenges, I'm pretty proud of separating almost 9,000 training recordings and plus like almost 2,000 Aki and like already over 2,000 Akepa. I listened to a lot of recordings, but I also learned the birds really well from that. And then also like, again, these transects are not easy. So transect 8A is like 6 miles, but you're like barreling through like really dense forest. So I'm also pretty proud that we all made it out like in one piece and got to hear some Akepa. So that was really cool. And for my ha'ina impacts, the song and call vocal phonology study is really like one of the first of its kind just because this technology is like relatively new and it allows us to engage with these bird songs so intimately. Again, like as Pat has mentioned before, like it's really one of the oldest languages we have in Hawaii. So really special to be able to work with them and understand them so intimately to help protect them. It really deepened my understanding of the ecology of native Hawaiian birds and contributed to research that can inform tangible actions for their conservation. And also, these spaces, like protecting these spaces is so important to maintain and help the upcoming generation develop Palina with native Hawaiian birds. I know there's a bird school up there which teaches people of all skill levels like about the native Hawaiian birds, and they can actually go out and see them up at Hakalau. I know Kaliko's group, Pilina Aina, took a group of kids up there so they can experience the birds too. So just so culturally significant too, like Leilani's Kahili, they mentioned the Kumulipo, and just, yeah. Also just seeing the bird murals everywhere, like we don't have that back in New Jersey. So really inspired by the connection that I've seen here with all the native birds. For next steps, I plan on following through on the song and call phonology study even after this summer. So in my dorm back in California, there will be the sounds of the Kepa and Amakihi and Oma'o playing. Warning to my roommate. We also might work on distinguishing types of songs and calls. We didn't do I'iwi or Apapane just because they're so variable. I learned in the first week that if I don't know what it is, it's probably an Apapane. So we just call everything memes, but there's like recognizable patterns within that like category of meme. Like, so we might try to see like if the timing of those is different throughout the year and if that means anything. And there's also other relevant bioacoustic projects back at UC Berkeley. Like I know they're working on a sound hub for California's wildlife. So a lot of these skills are directly transferable. And this really contributes directly to what kind of kapuna I would like to be. I want to be someone who helps protect and nurture bird populations for future generations. So all these skills I learned are really helpful for that. I just wanted to say thank you to the Pipes Ohana. Thank you for welcoming me into this space in which I'm a guest. I've learned so much from all of you guys. And I know the connections that we've built will last far beyond just this summer. I just wanted to thank you, huge thank you to Hilo, Pipes, Mama Sharon, Kiana, Vai, Leinalu Hinano, the Lohe Bioacoustics Lab. Thank you, Pat, Aiden, Sammy, Nahi, Buck, Keely, Amanda, Anne. It's really cool to see like all of you guys so well established and what kind of ornithologist I can hopefully aspire to be in the next few decades. Thank you to NSF for funding this experience, and also Jetstream and Perch for all the like models behind the scenes. And yeah, thank you so much. Mahalo.
Questions for our bird nerd. Our other bird nerd has a question for you, Ethan.
So while you've been here and been listening to all of the birds, I was wondering if you had a favorite of all of them, and like a favorite bird, but then also like a favorite call. Like it doesn't have to be the same one, you know? Yeah.
Yeah, I love all of them, but if I had to choose, I think the Kepa, just because they're like literally, I think they look like Cheeto Puffs. Also like their song, like the, I think they sound like fairies. The little cross, a Kepa, I love them.
Awesome presentation. Thank you so much.
Thank you.
Other questions for Ethan?
Hello, Ethan. I also thought you had a great presentation. Working in the Kīhoʻihoʻi area and being surrounded by some of the birds you mentioned, it's, as someone that's not really as into birds, but I want to get into it, how can I make it digestible? Like how, because there's just so many all at once. What do you recommend for taking on the bird world?
Yeah, I think, yeah, it is like a little intimidating when you first start out, but like kind of before I go to every place, I try to learn all the birds there. I just like to use, honestly, I just like to use Quizlet. Like just learning the birds like by sight kind of helps lock their names and what they look like in my mind. And then after that, I try to move on to songs and calls. Like I try to listen to recordings, just kind of, when you know what they sound like, you kind of know what to look for, and it's easier to like pick them out within the soundscape. But also I feel like one of the cool things about birds is they're so like inherently tied to place. I feel like you kind of have to be in the field to like fully understand their songs. So if you're ever free and down to go birding, yeah.
Sounds like a plan.
Yeah.
Thank you, Ethan.
Any last questions, comments, maybe from the lab that's here?
Great job, Ethan. So you've been going through all of these calls and songs, and so like the song of an akepa, for example, has a certain pattern to it, right? But do you think that they're trying to identify individuals when they sing? Like, do you think that each individual has its own particular song characteristic, and that they know who they're singing to when they're in the forest, based on what you've seen, like based on the variability?
Yeah, that's a really hard question. I did meet someone at HCC who I think might be with the Lohe Lab, but he's a TCBES student investigating like if you can identify individuals by their songs, I think. So that's one potential avenue. Just from personal experience, I think it's kind of hard to pick out individuals in the field, but just personally in New Jersey, I do feel like some individuals that like either are banded that you can recognize individually have different songs. Also just the fact that like birds, like it's kind of like both an inherited and an acquired skill that they learn to sing. Like there's obviously like something that's passed down genetically, but also like they learn a lot of the songs from like what they're hearing around them. So I think with that individual variability, I think it's totally possible. But yeah, I think definitely an area of future research. Yeah.
Amazing work, Ethan. We can't wait to see all the rest of the work that you have committed yourself to. And I think your excitement for the birds has definitely rubbed off on the entire cohort.
Yeah.
Can we get one last round of applause for Ethan? And up next, we have Miss Sara Oliveros with USDA going to tell us a little bit more about insects. So watch out, there might be some creepy crawlies on the screen.
Aloha, everybody. My name is Sara Oliveros, and this summer I worked with the U.S. Department of Agriculture, Tropical Pest Genetics and Molecular Biology Research Unit. I worked alongside Dr. Chan Hugh and Charles Mason. So a little background about me. I'm from Dallas, Texas, from the Blackland Prairie region. And today, less than 1% of the Blackland Prairie remains. This is 12 Hills. That's a picture of me from last year when I graduated high school. And it's a local pocket prairie in my community that is focused on Blackland Prairie restoration. And it was very involved in my initial environmental stewardship and education since elementary school. Since then, I gained the opportunity to become a junior master naturalist, be a part of the Trinity River Crew, and the Slingshot Challenge with the National Geographic. And being a part of the Slingshot Challenge was ultimately a full circle moment for me, where I shared the story of the Blackland Prairie and 12 Hills and gained an overall broader perspective and network of conservation and conservationists throughout the world. Through all these experiences in conservation, I learned the importance of protecting your Aina and its ability to protect and reflect its culture. This reflection of cultural identity, restoration, and community guided me to PIPES, where these values strongly lie within the program. With my project this summer, I gained experience in lab researching, data collection, and overall problem-solving skills to help combat the current environmental issues facing my own community. So at my research unit, the purpose is to develop new pest management technologies and enhance biocontrol agents, ultimately protecting American agriculture from invasive tropical pests. This would be probably a good time to look away if you're like squeamish. So my research this summer has been focused on Ceratitis capitata, the Mediterranean fruit fly, or medfly. And it's an invasive pest native to sub-Saharan Africa. Medfly is now established in here in Big Island, Waimea, on just low elevation farms and on Oahu as well. And there are occasional outbreaks on the mainland. The most recent one last week being officially declared eradicated in Santa Clara, California. So medflies pose a huge threat to agriculture because they lay their eggs inside fruits, nuts, and vegetables. And so when they hatch, the larvae feed on the fruit, making it unfit for consumption and for commercial use. And I'm sorry, this picture is like gross. And so yeah, we use sterile insect technique, SIT, and it's used to control pest infestations through the mass ring of male flies, then sterilizing them through gamma radiation, and then releasing them to mate with female flies, ultimately producing non-viable offspring. So RNA interference is a process that regulates gene expression, also known as gene knockdown. And I'm using it in my project. I'm researching its efficiency for SIT, potentially being used for things like sex sorting or as an alternative to irradiation. Currently, we use injection to mediate RNA interference into the medfly because it allows us to bypass digestive enzymes. However, this process can be extremely time-consuming. So my research question for this summer was, can E. coli be used to mediate RNA interference in Ceratitis capitata? And what are the optimal conditions for mediation? So beginning with my project, I synthesized my own double-stranded RNA. And to do this, I learned how to use sterile and safety lab techniques and common lab tools like pipetters, the vortex, and mini centrifuge. I also learned how to use the thermocycler and PCR for DNA amplification and how to create and run gel to make sure that my PCR worked properly. I also learned how to use tools like the qubit. I think Mina talked about it, which was just to check the concentrations of my solutions. And yeah, I synthesized my own double-stranded RNA because it allows me to make sure that everything is working properly and to choose specific knockdown genes with observable phenotypes. And so for my project, I chose, I'm using 3 different genes. So the first one is double sex, DSX. And this gene is used to disrupt sex differentiation and also affects egg-laying numbers. And the second one is ebony. And this creates a smokier, darker like coloring and markings on the wings. And also black pupa casings. So that's what this image is. In the center, you can see here the medfly. And this is the black pupa casings with the ebony knockdown, and then just like overall shadier colors on the fly. And the third gene that we're using is EGFP, and this is just a control because medfly does not have this gene, so we shouldn't see any physical phenotypes from this. And yeah, as I said before, the most efficient method of RNA interference right now in medflies is through injection. And then, like as I said, to bypass the digestive enzymes, because this would not allow the phenotypes to be maintained within the fly. So here's me performing some quality control injections. So I'm injecting pupa here, and they're super tiny, and so I used a tool called the NanoJect tool and a needle puller for injecting the double-stranded RNA. And so for this, I did several rounds of injections on larvae, pupa, and adult medflies. I did 30 larvae, 30 pupa, and 40 total adult flies, 20 male and 20 female. And I did this with each of the 3 genes. So I used the needle puller to create a small enough needle point that can penetrate the flies at a microscopic level to make sure that it doesn't harm them when they're getting like stabbed, I guess. And I used the nanojet tool to inject a consistent amount of solution into the flies. And what I learned through several hours of injections is that it is extremely time consuming. So one round of injections, like for each developmental stage, it was a minimum of 300 total injections. And so this would kind of take me all day to do. And things that also were setbacks was that the needle I was using was fragile, and the flies themselves are pretty fragile. So there was times where I had to replace the needles or replace the flies. And yeah, so that's why we want to use E. coli instead. And so before starting my project, I had to learn how to transform E. coli. So I did this with the plasmids, and I just made an LB broth and plated them into Petri dishes. And after this set, I created spiral plates with a transformed E. coli for it to grow. And so these are just examples of some of the plates we had. And yeah, I did. So I had 9 different constructs. So I used like 9 different cages. And these are just the constructs and their associated traits. So each gene had 3 different constructs. And some of them contain ZS green 1, which is a fluorescent protein that it helps us see through the fluorescent microscope if the E. coli has established. And this is the actual inoculation. So for the inoculation, we created 18% sucrose mix with the bacteria and set up pupa in there so that when they emerge, they'll feed on the solution, and that's how they become inoculated. And this is different for like the eggs and the larvae, because for those we used a diluted solution and poured it over them for them to feed on as they develop. And so this is how I analyzed my results. So I learned how to do dissections on the flies, and I dissected the abdomen to view ovaries and testes in the female and male flies. And I was looking to compare DSX and the control, and I'm basically looking to see if there's a difference between the color or the shape or the size of the sexual organs, and also if there's maybe like some flies have both sexual organs, ovaries and testes. Another type of dissection I did was a gut dissection, and that was used to view under the fluorescent microscope to see if there's fluorescence there. But our results, here's just an example of dissected ovaries from one of the med flies. But our results from the dissections of the organs was that there was no significant difference between the flies injected or inoculated with double sex gene and the control. Another way that we analyzed the data was a dissection of the individual gene experiments. So the left is ebony, and on the right side is EGFP. And as you can kind of also see from here, there's no significant difference between the markings or the colors of the wings. Wait, let me read this. And yeah, a little cat celebration, because we did get some positive results. So through the gut dissections, we were able to see the fluorescent protein. And this just lets us know that the bacteria was detected and established in the gut, and that the E. coli maintained itself and the plasmid was being retained. So I mean, you can see it from the fluorescence for the EGFP compared to the control, which had no fluorescence. And just something to note was that it's important for these situations to have a control, because sometimes things will have autofluorescence, so it's important to have a control there. And these are just more results. So these plates were made with homogeneized flies, and we just put it under a different microscope, and we had the same results, which was that it fluoresced. So just lets us know again that the E. coli has established. And so these are just some of the future directions of my project. So we'll continue doing like ongoing larval inoculations, just to see again like which developmental stage can be most efficient. Fine-tuning the injections, because we should have seen physical phenotypes from that, so we just need to figure out why we didn't see them from the injections. And maybe now with what we know about how we saw that the E. coli was established in the gut, we can try to target a different gene that may show an actual physical phenotype. And yeah, so this is just my future directions. I'm going to continue learning and leading. I have just finished my freshman year at Alcorn State University, where I study agriculture science with a concentration in environmental science. And at school, I plan on using the skills that I've gained this summer to support the conservation of the Mississippi Delta. And also, just like from what I learned in the lab, perseverance and stuff, I'll be making a comeback to this upcoming soccer season after an injury. So just using that mentality that I learned and applying that in my athletic career. And when I return to Dallas, I plan to apply these experiences in the lab and with PIPES to conservation needs in my own community. A big thank you to all of the sponsors who made it possible for me to come here, and especially thank you to Dr. Chan, Dr. Charlie, and Nick for their encouragement and dedication to my advancement this summer. This was my first time working in a lab, so like I can't emphasize the amount of patience and time it took to familiarize me with the scientific terminology, tools, and overall content of this experiment. And I'm excited to use these skills that I've gained throughout the summer. Mahalo to PIPES for allowing me to have the opportunity to explore Big Island and learn important parts of Hawaiian culture and history, and connect with so many amazing conservationists, and learned what it means firsthand to aloha aina. And then finally, hi to my family watching from home. No sería nada sin sus sacrificios, su apoyo y su amor. Los quiero mucho y los extraño mucho. Nos vemos pronto. Thank you.
Amazing, amazing job, Sara. Do we have any questions from the audience for her? From Sema.
Thank you for a great presentation, Sara. My question is, does the fruit fly, are there any specific crops that it targets, or is it more on like a wide array of different crops that it feeds on and we should be concerned about?
Yeah, thank you. And it's a wide array. There's lots of different vegetation that it targets. What I learned, I got to visit germplasm, and what I learned from there is that there are fruit flies that target the guava there. But yeah, medflies, they target all types of vegetation, avocados, like any kinds of fruit, really. Thank you.
Any last questions? If not, can we get a round of applause for Sara? Now, rounding us off for our very last presentation of the 2026 PIPES Symposium, our returnee, our bisque baddie, Miss Taylor Jenkins.
Aloha mai kākou. O Taylor Jenkins ko inoa, no manoa oahu mai au. This summer, I've been working with the Big Island Invasive Species Committee, or BISC, whose mission is to prevent, detect, and control the establishment and spread of invasive species threats to Hawaii Island's environment, economy, and way of life. My mentors are Darcy Yogi and Molly Murphy, both of which are PIPES alumni. They both wear a lot of different hats at BISC, including invasive plant prevention, CRB detection, and education and outreach events. So my project with BISC has been investigating the distribution, establishment, and fungal interactions of a non-native epiphytic orchid across Puna. And a little bit about myself. I'm a recent graduate of Colorado College, having majored in biology and ecology. And I believe as Kama'aina and as a student, that it is my obligation to give back to Aina for inspiring my curiosity for science for so many years. As I think about the kind of legacy I want to leave behind as I move on to graduate school and a professional career, it's just aloha Aina to the core. It really is my guiding star for a lot of the things that I do. And I may have graduated from Colorado College, but I'm still enrolled in Aina University. Oh, well, the formatting of this keeps changing every time I open up the story map, but basically, the conception for this project came from Molly Murphy, who had been observing and making observations on iNaturalists of several different genuses of epiphytic orchids across Puna and Hilo. And so I wanted to focus in on a single species of orchid, Epidendrum nocturnum, in order to understand whether or not it poses a threat to Hawaii Island. So some background on orchids. They're all in the family Orchidaceae, and there's only 3 orchid species that are endemic to Hawaii. And there's 14 known species or hybrids that have naturalized as of 2012, but that number has most certainly gone up since then. And a reason for this emerging trend of naturalization is the popularity of orchids in horticulture. You guys may have seen the AI-generated poster for the Hilo Orchid Show around downtown recently with the fairy. But that just happened this past weekend. And yeah, orchids are very popular here. But the reason that they're spreading is not well understood, and it comes sort of as a surprise because orchids tend to be underrepresented in invasive species. They don't have a lot of invasive characteristics, and they're considered ecologically specialized. One reason for this is that they require a fungal symbiont in order to germinate, every single one of them. And this fungus essentially inhabits the cells of the orchid seed and provides all of the carbon and mineral nutrients that the orchid needs in order to grow and germinate. And this relationship changes over time as the orchid matures. They might swap out their fungal partner, they might gather more fungal taxa. But the number of fungal taxa that they associate with can tell you about whether or not they might be considered a generalist or a specialist. That's an important point we'll get to because oftentimes 2 or more fungal taxa being associated with one species of orchid means that it might behave more like a generalist and it can colonize a variety of different habitats. And so the focus of this study is Epidendrum nocturnum, or the night-scented orchid, which occurs in Florida, West Indies, Central America, and parts of northern South America, and it is a self-pollinating plant. And so I did a little bit of literature review, and I came up with these predictor variables that I thought were most ecologically important and feasible for the PIPES timeline. And they include bark surface texture characteristics like rugosity, so how rough the bark is, how much it's peeling, and how much has actually fallen off of the tree. I'm also looking at canopy openness, which is essentially how much light or how much sky is open to the point where this orchid may have germinated on the tree. And then host tree diameter, breast height, is a metric for how old the tree is. And then we got elevation and rainfall data post-study to analyze as well. And so I went in two directions for this study. I wanted to take data on both orchid abundance and orchid recruitment. And these are related variables, but they tell a slightly different story. So orchid abundance is kind of just how many orchids are there on this one host tree, including both the mature plants with reproductive structures and seedlings. And then orchid recruitment is more of like a shorter timeline capture of orchid growth. So how many seedlings are there around a mature plant that may have just started spreading its seed? And then this is one of the data sheets that I made, one of several iterations that we used in this study with all of our variables. And so I designed two different studies for the summer, one being more focused on recruitment. There's also an element of rapid ohia death in this study because Molly had been making a lot of observations that these orchids were growing on dead ohia that had been impacted by rod. So when they die from rod, the crown will start to fall off and the bark starts kind of peeling from the top down, and it just kind of looks like this bare, sad tree. And you might notice like a ton of orchids just in full sun exposure at the top. And so I wanted to look into the association with orchid recruitment and the level of rod impact in ohia forest. And so my hypothesis for this part of the study was essentially that there's going to be higher recruitment in rod-affected ohia forests due to changes in bark surface texture and canopy openness that are caused by the death of these trees. So higher or larger or more rough bark and non-peeling bark being the variables that are more conducive to orchid recruitment. And then for the second study, it's more of just we're sampling at a lot of different sites, but we're asking whether these bark surface texture variables and canopy openness influence the abundance of this orchid. And also, what host trees are they being found on, and what orchid mycorrhizal fungi, or OMF, do they associate with? And then similarly, I expected that more rough bark and non-peeling bark, as well as high canopy openness, would correlate with more orchids. I'm also assuming that these orchids associate with two or more orchid mycorrhizal fungi because they've been seen to establish in a lot of different habitats. So they're likely behaving more like a generalist as far as their fungal symbiote tendencies. And then here's a map of the places that we sampled at. The yellow markers are for the abundance study, and then the red markers were where we collected our recruitment data. And so Steinbach Highway is considered a heavily rod-impacted forest. We sampled all along the road. And then the non-rod impacted forest, it just has little to no rod detection in that area, although rod is pretty prevalent all across Puna. We sampled here because it sort of had a matched up environmental variables with the Steinbach site, and we wanted to pick two sites that were sort of matched together in environmental variables so that the site didn't have such a large effect on recruitment alone. Yeah. So here's some photos from data collection. Me in a tree with Darcy helping me. Root collection. Making potato dextrose agar plates to grow our fungus. Doing surface sterilization of the roots. And then confirming the presence of pelotons, which is a specialized structure of OMF, where they basically inhabit the cells of the orchid root. And we actually have to extract them out and wash them and then try to grow them on plates. And here's Michael. Michael is kind of an unofficial mentor for this project. He's a third-year PhD student at the Hinson Lab, UH Manoa. And he helped us with this protocol. We sort of adapted one of his lab protocols from his work. And we actually had 12 plates of stuff growing, and we'll talk more about that. And so this is a cumulative results table for all of the different variables that we tested, and we actually do have some significance. So with bark rugosity, we found that there was higher orchid recruitment in the rough bark, as you can see in this graph. And for bark peeling, we see higher recruitment in the more peeled bark. And so we didn't find any significance with the canopy openness or the percentage of bark falling off. And there's also no difference between the rod and non-rod sites. The p-value is kind of close, though, so it could be a subject for future exploration. And then we also got a significant correlation, a negative correlation with elevation and rainfall. So here is the negative binomial distribution regression, where you can see that higher elevation and higher rainfall are associated with lower orchid recruitment. And then I wanted to do generalized linear modeling. It's a very effective tool in ecology, but not for this study because a lot of our variables are correlated with each other, which tends to make model outputs unreliable. And so this is the cumulative results table for the second study, the abundance study, and the results are a little bit different. We don't see the same significance for bark rugosity or bark peeling, but we do still see a negative correlation with elevation and rainfall. Oops. Yeah, visualizing that, higher rainfall, higher elevation is associated with a lower abundance of orchids. And then here is a pie chart mapping out all of the different orchid or host trees that the orchid was found on. And as you can see, there's one that is absolutely dominating the pie chart, and it's Hilo. And we actually have only one observation for this tree, and it had over 170 orchids on it. And this is a photo from just one part of the branch. It was a very large tree, but as you can see, there's like, these are all little seedlings. There's like at least 20 or 30 just on this branch. And so we were probably guesstimating well below how many were actually on the tree. But normally we only saw like a handful of orchids on each tree. And so this was pretty surprising. It could be, I don't know what about Hilo these orchids like so much. And so this is just the same pie chart, but with Hilo removed so that we can visualize some of the other species. We have hapu'u, different types of palms, mango, and ohi'olehua. And so on to the fungal part of this project. So like I showed earlier, we managed to culture out the pelotons, and we had 12 plates with lots of things growing on them, like this little white blob here. And so everything that kind of looked like it might be an orchid mycorrhizal fungi, we took a little bit of it, and we ran PCR with the ITS primer, and then we did DNA sequencing. And so we had 29 samples where we collected from our plates. And actually 14 of them came back with bands when we visualized our PCR product with gel electrophoresis, meaning that there's a hit for a fungus, essentially. And so when we sequenced these, we took the nucleotide sequence and ran it through the NCBI database, and we found a couple hits in these fungal taxa families, which are actually known orchid mycorrhizal families. So Telesnelaceae and Serendipitaceae are known to associate with other orchids globally. And so when we blasted the sequence, we found a couple other species of orchids that were native to Brazil for this sample. And then for our second sample, there were a couple orchids native to China, although the sequence similarity was a lot lower. It's kind of below the standard threshold, meaning that either our sample was kind of bad or no one has ever sequenced this species of fungi before. And so with the help of Michael, I made this phylogenetic tree, which essentially just puts into perspective the biogeographical or evolutionary history of our sample. So the sample that we sequenced is highlighted in yellow. And then these monophyletic groups that are all from the same location are highlighted with a color. We have Ecuador, Brazil, and Australia. These are all reference genomes from Michael. And what this is sort of telling us is that Brazil is closer to our sample, and potentially Brazil is closer in the biogeographic and evolutionary history of the sample. So maybe Brazil was in Brazil before it got to Hawaii, potentially. Or at least, it's part of the story. And then here is the tree for the second sample. The China origin orchids are a little bit closer to our sample here. But yeah, for my hoʻina, essentially these study results form a foundational understanding or a ka'au for the study species and its potential impacts on Hawaii Island. And like I said, bark rugosity and bark peeling had significant effects on orchid recruitment, suggesting that these variables might be promoting the germination and establishment of the species. But it's important to note that bark peeling in this study was mostly in the context of ohia, and so that might not produce the same effect in other host trees. And then elevation and rainfall were also significant negative predictors of orchid presence in both of our studies, meaning that they might be hindering the optimal conditions for germination and establishment of the species. Another thing is, since we're seeing slightly different results between the recruitment and abundance study, there might be something to say about which one is a better representation of current environmental variables. Recruitment might better explain seed capture and establishment compared to abundance, which is more of a cumulative measure over a longer timeline. And then more broader impacts. The spread of this orchid is still extremely understudied. And so one potential impact is that with this orchid spreading, it could facilitate the spread of other epiphytes because epiphytic plants are known to share their fungus. So if this orchid is establishing on a tree, a nearby plant might share the fungus with it, which could help it establish itself. And so another broader impact is that this information will go into the Hawaii Pacific Weed Risk Assessment System, which helps to provide risk scores for species based on their invasive characteristics and the availability of literature. This is an accessible system to the public, and it encourages informed decision-making in horticulture and landscaping. So hopefully, this information will go somewhere to help people perhaps not grow this orchid for horticulture. And then a last point is that while we didn't find any significance between the rod and non-rod sites, this is still potentially a foundation for further exploration on the connection between rapid ohia death and epiphytic orchid or other epiphyte establishment. And on that note, ohia lehua is an important mea kanu on Hawaii Island, and it's a kinolau of Hi'iaka, who has been a thematic figure of the 2026 PIPES program. So here's my acknowledgements. Darcy, thank you so much. You really, I couldn't have done this perhaps overly ambitious project without you. And I know I certainly took away a lot of your time from your deliverables and work and whatnot, but you're always down for everything that I wanted to do for this project. So I really appreciate it. Molly, Dustin and Chris from BISC, and Franny at BISC, who is a huge advocate for PIPES. Michael helped with all our fungi things. Hannah from the Ilala lab did all of the things in that lab. And then the PIPES Hui. Thank you guys so much. Oh, and then here's some bonus photos on the Mauna. This is me. I took a nap at Hui Mau, and then there was a bunch of photos of me that I found afterwards. Anyways. But yeah. But thank you guys so much for a great summer. I really appreciate it. Okay.
Great job, Taylor. Do we have any questions for her?
Hi, Taylor. Great job. I was wondering if the orchid mycorrhizal fungi symbionts that you found on these orchid species, are they native to the Hawaiian species of orchids as well? Or did they arrive with these non-native orchid species, and is that contributing, or if they are native to here, then is that contributing to this non-native orchid species ability to spread?
That is a really good question. That's actually not really known at this point, but Michael Fernandez, who's helping with this project, his PhD is actually comparing the orchid mycorrhizal fungi between native and non-native epiphytic orchids, so we actually probably will have an answer to that question soon. And one implication or application of his project is that potentially the invasive orchids could be used to help propagate native orchids if they're sharing the same fungi. But whether or not they're native is not well understood yet.
Any other questions for Taylor? Okay, cool.
Uh-oh.
Hi, bro.
Hi, bro.
So, so I was wondering, because I know obviously you work with a lot of invasive species, and this might be a weird question, but I was wondering if you have a favorite invasive species and why.
That's probably a good question. I mean, it's hard to like invasive species because they're bad. They don't belong here. But some of them are really beautiful, like orchids. They're all very beautiful. But I would say the bamboo orchid, I know like it's hated, but every time I see it, it's beautiful. It's all over the Volcanoes National Park. You guys have probably all seen it. But yeah, it's pretty, but that's about it.
So I have a question for you, Taylor. Just as a preface, I think one of the many things that the staff get to watch is like how people grow and the things they find that they didn't expect to find and the pathways that they do going forward. So it's been really fun to watch Taylor as she's done so many different things with so many different people in so many different places. And I've just always been impressed you just do it, and that's really great. So my question is, given all the experiences you've had and the places you've been, where does it take you now? Like, how has this influenced your pathway, and what are you going to do next? Because, this was kind of like almost PhD level stuff. Like wow, I was looking at how many questions did you have in this short a time? So yeah, what are you thinking?
Yeah, I mean, I feel like this is my second year of PIPES, and I kind of did like almost a master's thesis again, and it was very stressful last time, and it was very stressful this time, but I'm glad it really got done this time. But Justin is mad that I'm still working on the paper from the last PIPES year. But anyway, my future outlooks, I mean, I'm thinking about UH Manoa for graduate school. I want to take some time off, but I'm considering entomology, botany, but really like any field that needs me the most, I think. I'm very curious about almost everything, and so wherever they need me, I'll be there, and I can work hard. So thank you so much, Sharon.
Can we get one last round of applause for Taylor? And I'm gonna ask that Taylor stay up there and the rest of the PIPES who come up. She's our last presentation for today, so we would like to close with our mele mahalo once more.
Before we close, I really need to mahalo Revlin Takayasu and Lori Bufil, our amazing fiscal team that are really the gas in the tank for everything that we're able to do. And again, mahalo to everybody online for tuning in today or yesterday or both days. We're so proud of this group of students and looking forward to the next chapter of their ka'au. Mahalo. That's all we have, guys. Thank you. Thank you so much for your support of this program. Looking forward to next summer. Mahalo. Break. Interns are staying here, helping us clean up.
We would